So what we're going to do is we're going to do a live discussion on each topic. Let's say, I'll give an example. On September 1st, imagine that we're going to do anatomy, okay? So we are going to do anatomy from morning around 8:39 to evening 6:37 in 2 sessions. The morning will be the live discussion of all the questions of these high-heeled, hot competency areas of INI-CET going in depth. And then we'll stop it by evening, around 2 more hours left as a second session, where we'll take all the images alone asked in the last 5 years in INI-CET and the entire subject together and discuss the images alone to give you a focus on image-based questions and the applications around these topics. So it will be 2 parts in the bootcamp. One is the hardcore competency review of the entire subject, connecting you to each and every topic, and then a second smaller session of discussing all the images of the particular subject. Understood? So that is what is done on the day 1, and then you'll get 2 days to study. Like after first day anatomy, 2 days to revise or 1 day to revise anatomy by yourself from your primary notes, so that you can make sure that you get the 130 base correct. Understood? If not, studying this high yield area alone will never get you INI-CET. You have to make your fundamentals and basics strong and revise everything up. It's very, very important. Understood? So you do that 1 day revision and then you write the anatomy exam on the second day evening in the Medico app. And the next day, let's say if it is dermatology, 1 day dermatology, same thing. Morning the competency review topic question discussion, evening the image-based question discussion. Next day, time for you to study by yourself, evening exam. Then it's 2 days medicine. 2 days we'll discuss all the systems like this. Morning we will discuss the competency review topics. After that, evening image discussions. 2 to 3 days holiday, and third day evening exam. So that you are having a revision track to come back to your original notes, understand these are the areas to study, focus and learn more or go deeper. Also review a quick review of all the images and topics in the subject and making sure you touch your base notes completely, so that you're going to improve from week on week. So how do we assess that? Every week there will be an INI-CET mock test happening in Medico app, which you can come in and take, so that you will understand your score is improving or not.
Good morning, everyone. Hope you all are doing well and your preparations are in full swing for the forthcoming INI-CET exam that's going to happen in November. I'm Dr. Kanimuri Sadasivam, your mentor for physiology, and today we are going to discuss some questions which is going to help you in your forthcoming INI-CET exam. Today's discussion is basically going to be in 2 parts. The first part we will have clinical based questions, because nowadays most of the questions, even if it is from a basic science subject, the questions are all clinical based questions. So in the first session we are going to have some clinical based questions. We are going to discuss these questions, how to approach this question and how to answer them. In the second half, post lunch session, we will be looking at some image based questions. Most of these images are going to be charts, graph, how to interpret and how to go about answering these questions. So now, without wasting any more time, let us go into our discussion. So in physiology, the first question what we are going to see today, we have a 50-year-old male patient who is presenting us with symptoms of fatigue, night sweats and an enlarged spleen. So just by even without going into the entire details of the questions, these symptoms itself is telling you or giving you a clue towards what is the abnormalities. Okay, so it is going to be a hematological abnormality, right? So there is fatigue, there is night sweats and an enlarged spleen. Now a bone marrow biopsy reveals high number of abnormal cells. Okay, so we have a hematological disorder with high number of abnormal cell. So most probably it is either going to be a lymphoma or a leukemia, right? Any one of this could be a diagnosis here. Okay, now they are doing further test. Further test shows that these cells are accumulating in the G2 phase of the cell cycle. Which of the following regulatory mechanism is most likely disrupted in these abnormal cells? So first we have to understand the question. So what are they asking? They are giving us an hematological malignancy. So the clinical scenario here is an hematological malignancy. Okay, so this hematological malignancy has resulted because there is a defect in this G2 phase of the cell cycle. So the G2 phase of the cell cycle is defective. Because of that, abnormal cells are accumulating in this phase. Now, what is the question they are asking? Which is a regulatory protein that plays a role in the G2 phase of the cell cycle? So, to answer this question, you should have a detailed knowledge about cell cycle. What are the different phases of cell cycle? What is a cell cycle? What are the different phases of a cell cycle? And what are the regulatory mechanisms and regulatory proteins that play a role in each of these phases? So if you know this, it is going to be very, very easy to answer this question. Now let us look at the options they have given. So now you are clear with the question and what they are asking. So first read the question once or twice and understand it. Find out what are they asking. Then look at the options. Okay. So now they are asking the basically what are they asking here? They are asking which is the regulatory protein that has that is not functioning properly in the G2 phase of the cell cycle. Okay, so four options are given: cyclin D overproduction, inhibition of CDK4 or 6, defective P53 gene, and increased apoptosis. Okay, among these four options, one options, even if you don't know the answer, you can easily rule out is increased apoptosis. Apoptosis actually prevents the formation of abnormal cells. Okay, so it suppresses the formation of cancer cells. So definitely increased apoptosis is not going to be the answer. Okay, so before finding out, so now you have already delineated and ruled out one. So we are left with three other options. All these three options are regulatory proteins involved in different stages of cell cycle. Okay, now we should know which among these three options operates during the G2 phase of the cell cycle. Okay, so for that, let us first look at the cell cycle. What is cell cycle? Okay, so what is cell cycle and what are the different phases of cell cycle? So what is cell cycle? Cell cycle is nothing but series of changes. Okay, it is nothing but series of changes that a cell undergoes. Okay, series of changes that a cell undergoes during its process of development and maturation and cell division. So during this process, what are all the series of changes that a cell is undergoing? That is called as cell cycle. So the cell cycle has different phases. So the first phase is a phase of cell growth called as G1 phase. So during the C1 phase, as the name very clearly indicates, it is a phase of cell growth. So the cell enlarges in size, prepares itself for cell division. So how is it going to prepare itself? It is enlarging in size, it is synthesizing the protein that is necessary, and it is forming different organelles. So all these functions are happening during the G1 phase of the cell cycle. Okay, so the regulatory proteins that are important during this G1 cell phase of cell cycles are the P53 gene, okay, and RB protein. Okay, RB stands for retinoblastoma gene protein. So both these proteins are the regulatory mechanisms that is available for the G1 phase of the cell cycle. Now from G1, it will proceed to the next phase, that is the S phase of the cell cycle. Why do we call this as a S phase? Because this is a synthesis phase. It is the synthesis phase, that is why we call it as S phase of the cell cycle. What is getting synthesized here? DNA is making its copies. Okay, so for cell division, you need two daughter cells are going to be formed, right? So synthesis of DNA happens here in the S phase of the cell cycle. So basically DNA replication happens during S phase. So during this phase, what are all the regulatory proteins that play a role? Cyclin E and CDK. Okay, then from S phase, the cell is going to proceed to the next phase, that is the G2 phase. This G2 phase is also called as the gap phase of the cell cycle. So in this G2 phase, what is happening? This is one of the, among all the phases, this is one of the most important phase where checkpoint DNA damage check occurs. So the DNA damage check occurs during the G2 phase of the cell cycle. Okay, so this is the phase where the cell is preparing for its next phase, that is mitosis or cell division. Okay, so what are all the regulatory proteins that is going to help here? P53. So our question, if you remember our question, they were asking about the G2 phase of the cell cycle. So in G2 phase of the cell cycle, what are the regulatory proteins? P53, cyclin B, CDK1. Okay, so in G2 phase you have P53, cyclin B and CDK1. Then from the G2 phase, it then proceeds to the next phase of mitosis. Okay, so mitosis is also called as the M phase. Mitosis is also called as the M phase of the cell cycle, where mitosis and cytokinesis happen. That is division of the nucleus and cytoplasm happens. Even this mitosis proceeds through various stages. The first stage is the prophase, followed by metaphase, anaphase, telophase. Okay, so this is a cell cycle. This G1, yes, G2 phase, they are together called as interphase. Okay, so now I think you know the answer for the question. In the G2 phase of the cell cycle, which are the regulatory proteins? It is the answer is defective P53 gene. Why not CDK4 and cyclin D? We just saw in the G2 phase it is cyclin B, not cyclin D, and it is CDK1, not CDK6. Okay, so here it is actually, it has to be CDK1 and it is cyclin E and not cyclin D. That is why the option here is defective P53 gene. Okay, is it clear all of you? I hope it is clear. And some more details about this P53 gene. We also know this P53 gene is called as a tumor suppressor gene. It is also called as a tumor suppressor gene. Why do we call it as tumor suppressor gene? P53 gene is a crucial tumor suppressor protein. Why? Because it plays a very important role in responding to any damages that happen in the DNA. If it finds even a single DNA that is damaged, immediately the P53 gene will get activated, and that is going to lead to apoptosis. And it is going to prevent the formation of, it will prevent the cell cycle from proceeding to the next stage. From G2, the cell cycle will not proceed to the M phase. I hope you are clear with this. So just to summarize again, the various phases of cell cycle actually proceeds. Many MCQs have been asked from this topic. So this is one just example. With this discussion now, I hope you will be able to answer any question that comes from cell cycle. So what are the probable questions that we can expect in cell cycle? What are all the different regulatory proteins? Just go through this tabular column once. What are the different regulatory mechanisms that have a role during each phase of the cell cycle? What is the function of each phase of the cell cycle? Questions can come from that, as well as what is the order in which the cell cycle proceeds. So the order in which the cell cycle proceeds is G1 phase, followed by G1, followed by the S phase, then G2, and then M phase. Sometimes certain cells are also there in the G0 phase. Cells that doesn't undergo cell division will be present in the G0 phase. Okay, the cell will have all other functions. It will be performing all other functions except for cell division. So that is G0 phase. So this is the order of cell division. Okay, so now with that we will proceed on to our next question. So what is the next question? What are they asking? Which one of the following statements about cellular organelle is or true in the context of their role in cellular pathology? So basically they are asking, among the four options that is given here, which is the proper function of that particular cellular organelle. Okay, so the first option they're talking about mitochondria. So in option A, what are they telling? Mitochondrial dysfunction can lead to disorders characterized by impaired ATP. If mitochondria is not functioning properly, ATP production is impaired. Is this true? And leads to muscle weakness. Is this true? Yes. What is the most important function of mitochondria? It is called as the powerhouse of the cell. Mitochondria is called as the powerhouse of the cell. Why? Because it's called as the powerhouse of the cell because of its role in ATP production. So this is correct. What about the next option? Lysosomal storage disorders are caused by defects in lysosome, are caused by defects in Golgi apparatus, leading to improper protein synthesis. Is this the right answer? No. What is the function of lysosome? Lysosome contains hydrolytical enzymes. Lysosome is called as a suicidal bag of the cell. So it is basically going to attach to the phagosome, releases hydrolytical enzymes and causes the death of the organism, right? So that is not the function. Then what about endoplasm? Next option is endoplasmic reticulum. Endoplasmic reticulum stress is linked to diseases such as diabetes and neurodegenerative disorders due to accumulation of misfolded proteins. Endoplasmic reticulum, we know. What is the function of it? We have smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth endoplasmic reticulum, rough endoplasmic reticulum. Smooth endoplasmic reticulum, importance is synthesis of lipids. And rough endoplasmic reticulum is synthesis of proteins. Okay. Protein folding also happens in endoplasmic reticulum, but processing, packaging of proteins are all mainly the function of Golgi apparatus. Then finally, the last option, ribosomal defects in the ribosome will primarily affect the DNA replication and repair mechanisms of the cell. Is this the right option? No, DNA replication is the function of nucleus, not the ribosome. Okay, so basically to answer this cell, what are the things you're supposed to, sorry, basically to answer this question, what are the content you're supposed to know? You should know about the cellular organelles, their function. Okay, if you know this, you will be able to easily answer this question. It is a very easy direct question. So what is the correct answer here? Mitochondrial dysfunction will lead to impairment in ATP synthesis, which is going to lead to muscle weakness. So that is the right answer. Okay, now to add more, let us also discuss some important information. This is a direct question. They have directly asked you about the cellular organelle in function. Okay, there are few more questions which they keep asking from this topic, mainly the enzymes that is present in these organelles, what is the function of these organelles, and also which is the marker for each of these organelles. So all this is important. Enzyme is an entire topic. It is very large. I've already covered it in our Medico app. You can always open the Medico app and see general physiology, where we have discussed that in detail, all the enzymes that is present in different cellular organelles, where it is present and what is its function. Deficiency of the enzyme is going to lead to what disorder. All those things are discussed there. Now here in brief, let us look at the cellular organelle, the marker for that particular cellular organelle and the function of that organelle. Okay, so we will start with nucleus. Just now we saw. So the marker for nucleus is DNA, and this is important for DNA synthesis. Okay, nucleus is important for DNA synthesis and repair. Next organelle is mitochondria. We just know we saw what is the function of mitochondria. ATP, primarily ATP synthesis. And what is the marker for mitochondria? Glutamate dehydrogenase. So the marker for mitochondria is glutamate dehydrogenase. Then coming to the next organelle, ribosome. The marker for ribosome is the RNA. Okay, and ribosome is a site of protein synthesis. Endoplasmic reticulum, the marker for endoplasmic reticulum is glucose 6 phosphatase. For endoplasmic reticulum, the marker is glucose 6 phosphatase. Okay. Coming to lysosome, the marker for lysosome is acid phosphatase. Lysosome has more than 40 hydrolytic enzymes. Okay. Sorry. Then comes plasma membrane. The plasma membrane, the marker for plasma membrane is the sodium potassium ATPase and the 5 nucleotidase. What is the importance of plasma membrane? It helps in transport of molecules in and out of the cell. It is important for intercellular adhesion and communication. Coming to Golgi apparatus, the marker for Golgi apparatus is galactosyl transferase. And Golgi apparatus is important for sorting out of proteins and also for glycosylation, that is attachment of carbohydrates. Okay, so both are function of the Golgi apparatus. Then coming to peroxisomes, peroxisome, the marker for peroxisome is catalase uric acid oxidase. And what is the function of peroxisome? It is protecting the cell against free radical damage. Then comes the cytoskeletal elements. We don't have specific markers. The cytoskeletal elements are mainly the microfilaments, microtubule and intermediate filaments. They help in movement of substances within the cell and also for movement of the cell from one place to the other. Then comes cytosol. The marker for cytosol is lactate dehydrogenase, and the enzymes that is present in the cytosol is important for glycolysis and fatty acid synthesis. Okay, so this is in brief the various functions of the cellular organelles and the markers for these cellular organelles. We'll just take a small technical break. Welcome back to the session. So there was a question in the live where they've asked about the function of 5-nucleotidase. So this 5-nucleotidase is an enzyme which is very important for the nucleotide metabolism. It's an enzyme for nucleotide metabolism, and it is also a marker for liver function. Okay, additionally, it's an important question. Thanks for coming up with the question. 5-nucleotidase is a marker of liver function. Okay, if this enzyme is released into the bloodstream, it is telling that there is liver damage. Okay, it helps in nucleotide metabolism, meaning it helps in conversion of ATP into adenosine. It helps in adenosine formation. I hope I have cleared your doubt. Now let's move on to our next question. Okay, sorry. We have a 40-year-old woman with a history of chronic liver disease, and she presents with ascites and jaundice. Okay, so we have a 40-year middle-aged woman with history of chronic liver disease presenting with ascites and jaundice. The laboratory tests reveal elevated levels of cholesterol. So please underline or make a note of these key points. That is what is going to help us answer our question. So we have a patient with chronic liver disease presenting now with ascites and jaundice. So it has progressed, the disease has progressed. And lab investigation reveal elevated cholesterol and phospholipids in her blood. Okay, this alteration in the lipid profiles can affect the cell membrane. So they are already telling you. So there is altered cholesterol phospholipid ratio, and this is affecting the cell membrane function. Which of the following is a likely consequence of this observed lipid imbalance in the cell membrane? Okay, so basically to answer this question, what are you supposed to know? The structure, composition, and functions of cell membrane. Okay, so one key thing what they are telling you in this question is because of the patient's pathological condition, because of her liver condition, the phospholipid cholesterol ratio in the cell membrane is altered. The cholesterol content has increased. So now they are asking what is the consequence of this increased cholesterol in this pertaining to the function of the cell membrane. What will happen to the cell membrane if the cholesterol in the cell membrane increases? And what will happen if the cholesterol phospholipid ratio of the cell membrane gets altered? So that is the question here. Before looking at the options, let us briefly discuss about the cell membrane, its functions. Okay. So here we have our cell membrane. Okay. So here we have the cell membrane. So you all know cell membrane is made up of, what is the composition of the cell membrane? The cell membrane is made up of lipids, protein, and carbohydrates. Okay. Now in the dry weight, how much percentage is contributed by all of these? Lipids contribute around 55 percentage. Okay, proteins contribute around 42 percentage, and carbohydrates contribute about 3 percentage. Or if we have to roughly say, proteins and lipids will be in the ratio of 1:1. Okay, so lipids are 55 percentage, proteins is 42 percentage, and carbohydrates contribute 3 percentage. Okay, now in this, what are the different types of lipids? Lipids in the cell membrane are of 3 types. Okay, phospholipids. Okay, okay, lipids are of 3 types. Okay, what are the 3 types of lipids that is present in the cell membrane? Glycolipids, that is what you are seeing here, a carbohydrate attached to the lipid, so that is called as a glycolipid. Then we have phospholipids. The bilayer of the cell membrane is basically made up of phospholipids, and then we have cholesterol. Okay, interspersed, so this is the cholesterol which is interspersed between this phospholipid bilayer. So this is a phospholipid. So if you look at the structure of the phospholipid, it has a globular head and a tail. Okay, we call this phospholipid as an amphoteric molecule. Why? Because it has both polar and non-polar region. The globular head is polar, the tail is non-polar. All these are MCQ questions. Okay, the head is polar, that is why it is facing towards the water. Why? The polar head means it doesn't repel water. It is hydrophilic head. Okay, hydrophilic. Polar means hydrophilic. Then we have the tail region, which is hydrophobic, non-polar. So the cell membrane is basically made up of a lipid bilayer. What you see here is a lipid bilayer. Okay, this lipid bilayer is made up of nothing but phospholipids. So this lipid bilayer is made up of phospholipids. The phospholipids have a globular head and a tail. So since this tail is hydrophilic, it is not going to face water. So when you look at the cell membrane, the outside the cell membrane you have fluid, that is the extracellular fluid. Inside the cell membrane you have the intracellular fluid. So it is the globular head will be positioned in such a way that the head faces the extracellular fluid as well as the intracellular fluid. That is why we have a lipid bilayer. Okay, in between this bilayer you have the tail region which is facing each other. So that is the phospholipid bilayer. This phospholipid bilayer is also called as the fluid mosaic model of the cell membrane. Why? Because the cell membrane is not rigid, it is in the fluid form. It is in the fluid form, yet it maintains the structural integrity of the cell. Okay. And why is it in the fluid form? Because of this particular arrangement and presence of lipids. Now, interspersed, already I told you, glycolipid loosely attached to the globular head on the extracellular. This is extracellular fluid, it is intracellular fluid side. On the extracellular fluid side, ECF side is our attached to the extracellular fluid side is the glycolipid. Okay, and then what else? Three types of lipids are there, I told you. Already we saw about glycolipid, we saw about phospholipid. Now the third is the cholesterol. Where do you find cholesterol? Interspersed between the lipid bilayer is the cholesterol in general. Okay, in general, in a normal cell, the phospholipid cholesterol ratio, okay, the phospholipid cholesterol ratio is phospholipid cholesterol ratio is 1 is to 1. Okay, normally it is 1 is to 1. So in our question, what is happening? Now I hope the cell membrane structure is clear. So now in our scenario, what has happened? The cholesterol has increased. Okay, and I told you it is because of this phospholipids arranged in this particular pattern, we have the fluid mosaic model. If you increase the cholesterol, if we increase the cholesterol, what is going to happen? Look here, this is a cell membrane with no cholesterol. It is fluid in nature. So when these cholesterol molecules come and attached in between this phospholipid layer, what is going to happen? The rigidity of the cell membrane gets increased. The rigidity of the cell membrane increases. Okay, so that is what they are asking us in the question. What is the consequence of increased cholesterol? Increase in cholesterol in the cell membrane is going to make the cell membrane more rigid. That is the answer. Okay, now what are the other factors that affects the fluidity of the cell membrane? So we already saw, if there is no cholesterol interspersed in between it, it is going to be more fluid. What are the other things? Temperature. Increase in temperature is going to increase the fluidity. Okay, and then type of cholesterol. Okay, type of cholesterol or type of fat is also important. Okay, we have the polyunsaturated fatty acids and saturated fatty acids. Okay, polyunsaturated fatty acids will increase the fluidity, whereas saturated fatty acid is going to decrease the fluidity of the cell membrane. So these are all the factors. So not only they can ask you the consequence of cholesterol, some other direct questions like this can also come. Among the following, which of the one will not affect the fluidity of the cell membrane, or which of the one is affecting the fluidity of the cell membrane? So any questions they can ask. So the fluidity of the cell membrane will depend upon the cholesterol, temperature, and the type of fat that is present. Okay, so now with this, let us approach this question. Let us look at the options. So Which of the following is a likely consequence of the observed lipid imbalance in the cell membrane? Option A, enhanced membrane fluidity and permeability. Definitely no. The membrane fluidity, if cholesterol is increased, the membrane, it becomes more rigid. The fluidity will decrease actually. Decreased receptor mediated endocytosis. Receptor mediated endocytosis type of transport mechanism. So that is not the option here. Increased membrane stability and reduced fluidity. Yes. So when you add cholesterol, the membrane is going to get stabilized. At the same time, its fluidity will be lost. Okay, it is becoming more rigid. Impaired ion channel function, no. So the answer for our question is, if cholesterol in the cell membrane increases, the cell membrane is going to become more rigid and it is going to lose its fluidity. Other things that affects the fluidity of the cell membrane are the type of fat. Polyunsaturated fatty acids makes it more fluid. Saturated fatty acids is going to make it more rigid. Temperature directly proportional. Increase in temperature is going to make the cell membrane more fluid. Decrease in temperature is going to make the cell membrane less fluid. Okay. Okay. So coming to the next question. Before reading the next question, because this question is also about cell membrane, let us finish our discussion on cell membrane and then we will come back to this question. So where were we? We saw the composition of the cell membrane. We saw the composition of the cell membrane and the ratio of phospholipids and cholesterol in the cell membrane. In general also, the ratio of protein to fat in cell membrane is around 1:1, equal amount, 50% of protein, 50% of lipids. But this ratio of protein to lipid can change depending upon the type of cell and the function of that cell. For example, so this ratio of protein lipid ratio in different cell membranes. So if we look at a Schwann cell, if we look at the Schwann cell, what is the most important function of the Schwann cell? Myelination, right? For myelination, what do you need? Proteins or fats? We need fat. Okay, so in such a cell, the fat is going to be more. More than 75% of the cell membrane is going to be made up of fat, and only 25% is going to be protein. So the ratio will be very less. The ratio of protein to lipid is going to be 0.23. That is, the protein is less, the fat is more. Whereas if you look at the inner mitochondrial membrane, the ratio is high, 3.2. Why? Because mitochondria is a metabolically active cell. It is a powerhouse of the cell. It is producing ATP synthesis is happening here. So in such cell, you need lot of proteins. So the protein is more and the lipid is less. Okay, so inner mitochondrial membrane, the protein to lipid ratio is high, whereas in a Schwann cell, the protein to lipid ratio is low. Okay, so depending on the cell and their function, the protein to lipid ratio can vary. But in general, it is 1:1. Okay, so if they ask you a question, which has the highest protein to lipid ratio, what will you say? Inner mitochondrial membrane. Which has the lowest? The Schwann cells. Okay, so now from the lipid bilayer, we are moving on to the substances that is embedded in it. So we discussed about the lipids. Then we are moving on to the proteins. Okay. So here in this lipid bilayer, where do you see proteins? Proteins can be present on the outer surface of the cell membrane. Proteins can be present on the inner surface of the cell membrane, or proteins can span the entire width of the cell membrane. Depending upon where they are located, they are named differently. Okay, let us look at these proteins. Okay, the proteins that is present on the outer membrane of the cell membrane is called as extrinsic protein. Protein. The proteins which are present on the inner surface are called as intrinsic protein. Okay, the protein that spans the entire cell membrane is called as transmembrane protein or integral protein. It's called as integral protein. Okay, so integral proteins. Now let us look a little bit about integral proteins. Okay, the integral proteins are also called as transmembrane proteins. These integral proteins have several important functions. Okay, one, it serves as channel proteins. It acts as carriers. It acts as ion pumps. It can also act as receptor and enzyme proteins, and these are also antigenic in function. So these are all functions of integral protein. An integral protein could be a channel like a sodium channel, potassium channel, calcium channel. It can serve as a carrier like your sodium glucose co-transporter or glucose transporter. Any of this is a carrier protein. It can serve as ion pumps, sodium potassium ATPase pump, hydrogen ATPase pump, hydrogen potassium pump. So or it can also act as a pump. It can act as receptors for various hormones. It can act as enzymes and also antigenic function. The blood group antigen present on the RBC surface, these are all integral proteins. Okay. Now talking about peripheral proteins, already I told you, peripheral proteins are of two types, intrinsic peripheral proteins and extrinsic. Those facing the extracellular fluid side, those facing the ECF side is called as the extrinsic. Those facing the intracellular fluid is called as the intrinsic protein. So this intrinsic proteins, which is facing towards the inside of the cell membrane, these intrinsic proteins are usually enzymes or proteins for the cytoskeletal elements like microfilaments, which is going to maintain the shape of the cell. The peripheral proteins which are facing the outside of the cell membrane, okay, they mostly serve as cell adhesion molecules. They mostly serve as cell adhesion molecules, okay. So now I think we have briefly discussed about the cell membrane, its composition and function. Now let's look at that question. Okay. Which of the following statements about the cell membrane is true? I'm sorry. The cell membrane is primarily composed of a single layer of phospholipid with embedded proteins. Is the cell membrane made up of a single layer of phospholipid? No, it is phospholipid bilayer. So this is the wrong option. The fluid mosaic model describes the cell membrane as a rigid structure with fixed proteins and phospholipids. Is it a rigid structure? No, it is a fluid structure. So this is also not the right answer. Next, the cell membrane has a selective permeable nature, allowing only non-polar molecules to pass freely. It has selective permeability, that is true. But it is not that it only allows non-polar substances. It allows both polar and non-polar substances to pass through it, only the mechanism is different. So this is also not the right answer. So we are only left with option D. So what are they talking in option D? The cell membrane's fluidity is mainly due to the presence of cholesterol molecules that stabilizes the phospholipid bilayer. Yes, correct. The cholesterol molecules are the ones which is stabilizing the phospholipid bilayer. But only when the cholesterol increases, it makes it more rigid. So what is the answer here? Answer D. The right answer is the cell membrane's fluidity is mainly due to the presence of cholesterol molecules, and these cholesterol molecules stabilizes the phospholipid bilayer. Okay. Okay. Coming to the next question. Which of the following components of the extracellular matrix is primarily responsible for providing structural support and tensile strength to the tissues? Which of the following components of the extracellular matrix is responsible for providing structural support and tensile strength to the tissues? They have given four options: glycosaminoglycans, collagen, fibronectin, and elastin. All the four components given in the options have a role in the extracellular matrix. Okay, they are all part of the extracellular matrix, but in this they are specifically asking among the 4 options which is having the role of providing the tensile strength and structural support. Okay, directly it's a direct question and the answer is collagen. Okay, why? Because collagen is the primary structural protein in the extracellular matrix, and it is very, very important to provide the tensile strength and structural support that is needed to the tissues. Then what about the other options? The other options given here are glycosaminoglycans, fibronectin, and elastin. So what does it do? The glycosaminoglycans are mainly for water, hydration of the cell, hydration of the extracellular matrix. Okay, so it is mainly involved in providing the hydration and cushioning. Then what does fibronectin do? It facilitates cell to cell adhesion and helps in cell signaling. Fibronectin for cell to cell adhesion and cell signaling and cell migration. Then what about elastin? Elastin is important for stretching of the cell, elastic recoiling. For all those things you need elastin. So among the 4 options, so which is the one which provides the tensile strength and structural support to the extracellular matrix? It is collagen. So if they talk about hydration of the cell, then it is glycosaminoglycans. Okay, cell to cell adhesion means we will be talking about cell. If they ask cell to cell adhesion, then it is fibronectin. Okay, hydration and cushioning effect, then it is glycosaminoglycans. Recoil, elastin. For elastic recoiling, we need elastin. Okay. Okay. Now we have our next question. We have a 35-year-old patient diagnosed with a genetic disorder that affects the function of sodium potassium ATPase pump. So there is a defect in the function of sodium potassium ATPase pump is defective. This defect leads to an inability to maintain the normal electrochemical gradient across the cell membrane. So they have told because of the defect in this pump, the electrochemical gradient across the cell membrane is not maintained properly. Now what is the question then? Which of the following is a likely direct consequence of this defect? That is, because of not maintaining a proper electrochemical gradient across the cell membrane due to a defective sodium potassium ATPase pump, what is going to happen to the cell? That is what their question is. Okay, so what is the sodium potassium ATPase pump? Sodium potassium ATPase pump, where is it present? So here what you see is a sodium potassium ATPase pump. It is present in almost cell membrane of all the cells. It is an active pump. What is it going to do? What are the functions of this pump? One function, what is it going to do? Electrical and chemical gradient. It is important for establishing the electrochemical gradient. Cell volume. It is important for maintaining the cell volume. Okay. So where is we just now we saw where the pump is located. What is the function of this brief important function of this pump? Okay, how does this pump operate? It is going to pump 3 sodium ions outside of the cell from the inside. It is pumping 3. Can you see here? It pumps 3 sodium ions outside of the cell and 2 potassium ions into the cell. Okay, so what is it doing? 3 positive ions are leaving the cell. In return, only 2 positive ions are coming. So there is always a electrical gradient that is established. Okay, there is one the negative charge. There is more negative charge inside of the cell membrane compared to the outside. Why? Because we are losing 3 positive ions, but in return only 2 positive ions are coming. That is why inside of the cell membrane is more negative compared to the outside of the cell membrane. That is called as establishing an electrical gradient. Then how does it establish a chemical gradient? It establishes the gradient for sodium ions. Okay, by actively pumping 3 sodium ions from inside of the cell, that is from a region of lower concentration, it is pumping 3 sodium ions outside to a region of higher concentration. The sodium concentration is more on the extracellular fluid side, and it is pumping further sodium ions outside. So it creates a gradient for sodium influx. So that is called as chemical gradient. That is why electrochemical gradient is established by the sodium potassium pump. Okay, by establishing this electrochemical gradient, the sodium potassium pump helps in maintaining the resting membrane potential. Maintaining the resting membrane potential. The permeability of potassium helps in establishing the resting membrane potential. Then maintaining the resting membrane potential is mainly the function of the sodium potassium ATPase pump. And also by actively pumping sodium ions out of the cell, it helps in maintaining the cell volume. If more sodium ions are accumulating inside the cell, what will happen? The cell is going to hold more water, that is going to cause cell swelling, and finally cell will burst and the cell will die. So now all these things are prevented because of the sodium potassium pump. So now I guess you are clear with the functions of the sodium potassium pump. It helps in maintaining the establishing an electrochemical gradient. It maintains the cell volume by pumping out sodium ions, and it's also important for maintaining the resting membrane potential. So that is the function of the sodium potassium ATPase pump. Now let us look at this question. So the defect in the sodium potassium ATPase pump, because of this defect, what is the likely direct consequence? Option A, decreased intracellular calcium concentration. No. Option B, increased intracellular sodium concentration. Yes, just now we saw. It helps in maintaining the cell volume. Now when this pump is defective, what is going to happen? Sodium will not be pumped out of the cell. So that is going to increase the intracellular sodium concentration. So the right answer is option B. Okay. What about option C? Increased extracellular potassium concentration? No. Decreased extracellular sodium concentration? No. What is so? The right answer is increased intracellular sodium concentration. Okay. Now let us also briefly look at the structure of the sodium potassium ATPase pump. Okay, so this sodium potassium ATPase pump has an alpha subunit and a beta subunit. The alpha subunit is large, the beta subunit is small. If you look at the alpha subunit, you can see okay, so if you look at the alpha subunit, this alpha subunit spans the cell membrane 10 times. Very important. Okay, it spans the cell membrane 10 times. G protein is also called a serpentine protein. It will span the cell membrane 7 times. So most of the times we'll confuse and we'll the 7 is our favorite number. We'll tend to select 7. But when it comes to sodium potassium ATPase pump, it's a large pump. The alpha subunit is large compared to the beta subunit, and the alpha subunit spans the cell membrane 10 times. Okay, this alpha subunit has an extracellular domain and an intracellular domain. In the intracellular domain, the alpha subunit has 3 sites. One site is for attachment of sodium ion, the other site is for attachment of ATP, then the other site is attachment for the ATP phosphorylation site. Okay, so this is the phosphorylation site. That is the intracellular site. If you look at the extracellular site, it has 2 sites. One is for attachment of albumin, okay, and then the second is for attachment of potassium ions. That is with the alpha subunit. Some more points about the alpha subunit is alpha has 3 forms: alpha 1, alpha 2, alpha 3. Alpha 1 is commonly present in the sodium potassium ATPase, which is present in almost all the cells, whereas the alpha 2 isoform of this alpha subunit is mainly present in the skeletal muscle, heart, adipose tissue and brain. Okay, skeletal muscle, heart, adipose tissue and brain will have the alpha 2 isoform of the alpha subunit, and the alpha 3 isoform is mainly present in the heart and the brain. Okay, alpha 3 is in the heart and the brain. Alpha 2 is in the heart, brain, additionally adipose tissue and skeletal muscle. This adipose tissue skeletal muscle, you can also remember for GLUT4. So the same thing, skeletal muscle, adipose tissue, additionally heart and the brain. That is alpha 2 subunit. Alpha 1 subunit is present in all the cells. Okay, that is with regards to alpha subunit. So two things you have to remember when it comes to alpha subunit. It is a large subunit, spans the cell membrane 10 times. It has intracellular domain, extracellular domain. Extracellular domain, potassium and Hoban binding units are there. On the intracellular site, phosphorylation site is there, ATP binding site is there, sodium binding site is there. Okay, so that is with regards to the alpha subunit. Now coming to the beta subunit. Okay, this beta subunit is also having beta 1 and beta 2 isoforms. Okay, beta 1 is widely distributed everywhere, but it is absent. Here in alpha subunit, we saw where alpha 1 subunit is present, where alpha 2 and alpha 3 are present. In case of beta subunit, where it is absent, beta 1 is absent in certain astrocytes and vestibular cells of the inner ear and fast twitch muscle fibers. In all these three places, beta is absent. Okay, okay. Whereas fast twitch muscle fibers will contain only the beta 2 subunit. Okay, and this beta subunits on the extracellular site, they have binding sites for carbohydrate. Okay, so carbohydrate binding site is present here. Carbohydrate binding site. Okay, so that is with the structure and function of sodium potassium ATPase pump. Lot of questions have been asked from this about this pump. So if they ask you where does cardiac glycosidase bind, so we will be talking about the Hoban binding site. So it will be binding on the extracellular site. Okay. Now coming to the next question. Okay, did we answer the options? Okay, we saw the options. Okay, now the next question. Which of the following mechanism of transport across the cell membrane does not require the input of energy? This is the most direct question I have seen so far. Okay, they are talking about transport across cell membrane. In the transport mechanism, among the given four transport mechanism, which one does not use energy, does not use ATP? What is the answer? By now you should be able to tell what is the answer. Which one does not, which? So passive transport doesn't require energy. So among the four given option, which is a passive transport mechanism? Definitely option A, they have told us active transport, so that is not the answer. Look at option C, endocytosis. Endocytosis is a form of active transport because it requires energy. Endocytosis, exocytosis, all this vesicular transport, though they don't need a concentration gradient, but still they need metabolic energy. So it comes under active transport. Primary transport is also a type of active transport. So what is the answer? Facilitated diffusion. The moment you see the word diffusion, you know it is passive transport. Okay, so here we have a classification telling us different types of transport mechanism. Before that, we will briefly see the differences between active and passive transport. Active transport, passive transport. So as we already saw, active transport, ATP is required, ATP is not required. Okay, second thing, active transport mechanism will follow saturation kinetics. Okay, whereas simple diffusion, there is no saturation kinetics and no competitive inhibition. Okay, whereas in case of simple diffusion, none of this thing is required, whereas facilitated diffusion will follow saturation kinetics and competitive inhibition. Then active transport is uphill transport, that is against the concentration gradient. Okay, whereas passive transport is downhill along the concentration gradient. Okay, so these are briefly the differences between active and passive transport. So we will just look at this, what is the meaning of saturation kinetics and competitive inhibition. Okay, any transport, be it active or be it facilitated diffusion, whenever a carrier protein is involved, then it is going to follow saturation kinetics and competitive inhibition. What do we mean by saturation kinetics? The transport of that particular substance is not indefinite. Okay, it is going to get the transport is only going to happen until it reaches its transport maximum. That is called as saturation kinetics. Once all the transporters are saturated with this particular molecule that has to be transported, then no further transport of that particular molecule will happen. That is, after reaching the V max, transport maximum, no further transport of that particular molecule is going to happen. That is called as saturation kinetics. Then what do we mean by this word competitive inhibition? Competitive inhibition means we have a carrier protein or a transporter which is going to transport a particular molecule. The same transporter might also transport another similar molecule. For example, D-glucose and D-galactose. Okay, GLUT is going to transport both D-glucose as well as D-galactose. Now, when both are present, okay, when both are present, both of them are going to compete with each other for that particular GLUT transporter. So that is called as competitive inhibition. So competitive inhibition is a property of both active transport as well as passive transport. Specifically for when we are talking about passive transport, we are specifically talking about facilitated diffusion. Okay, these concepts should be clear because they can twist and ask you questions in this. Whereas ATP is definitely not required for passive transport. Okay, and passive transport is always along the concentration gradient, that is from a region of higher concentration to a region of lower concentration. Okay, so these are the very basics where you are not supposed to go wrong. Now let us look at the classification of cellular transport. Okay, cellular transport, just now we saw we have active transport and passive transport. Passive transport is again of different types, mainly simple diffusion, osmosis, facilitated diffusion. Other than that, we also have filtration, bulk flow, solvent drag. Okay, so all these six are examples of passive transport. Okay, the moment you see the word passive transport, what are we going to say? No ATP. Be very clear. Then we have active transport. Active transport is again of actually three types. One is with the help of pumps, the other one is vesicular transport, then transport across epithelia. Okay, transport across epithelia. Three types of transport are there, and this most important one are the using pumps. We have the primary active transport and secondary active transport. Vesicular transport, we have exocytosis and endocytosis. Okay, and then transport across epithelia is also a type of active transport where we have transcellular transport and paracellular. Okay, transcellular and paracellular is very easy. When a substance is transported through the spaces in between the cells, we call it as paracellular route. Okay, when a substance is transported through the cell from the basolateral membrane, it enters the cell and goes into the interstitium or enters into the blood. We call it as transcellular through the cell. Okay, can you think of any examples of transcellular? All these nutrients, glucose is absorbed from the lumen of the gut into the epithelial cells. From the epithelial cells, it is again will be transported into the bloodstream. So that is all transcellular route. Paracellular route, certain ions, water will move, not water, water goes through aquaporins. Ions are going to go through these paracellular routes also. Okay, now coming to active and passive transport. Okay, so let us look at, first let us look at this active and passive transport. First let us look at the passive transport. Simple diffusion. What do you mean by simple diffusion? So you have two pictures here. Can you identify which is simple diffusion and which is facilitated diffusion? Yes. So what you see here, the first one is simple diffusion. And the second one here is facilitated diffusion. Okay. Why is this simple diffusion? Because you can see the substance is moving from the region of higher concentration to the region of lower concentration by simply passing through the lipid bilayer. What are the substances that can move through this? This could be gases. It could be alcohol, substances that are lipid soluble. Okay. And coming to facilitated diffusion, what do you see here? Again, from a region of higher concentration to the region of lower concentration. Okay, but what is important here is with the help of a carrier protein. Okay, with the help of a carrier protein. This carrier protein is the one which is transporting this particular substance from outside to inside of the cell. So this type of transport is called as facilitated diffusion. Example of facilitated diffusion is glucose transporters. Okay. So this is facilitated diffusion, which is again example of passive transport. Now we are left with other types of mechanisms. What are those? Osmosis. What do you mean by osmosis? Osmosis means movement of solvent from a region of lower solute concentration to a region of higher solute concentration. Okay, so we have this is a semi-permeable membrane. Okay, so when water is moving, okay, when fluid is moving from a region of lower solute concentration to a region of higher solute concentration, when fluid is moving from a region of lower solute concentration to a region of higher solute concentration, we call it as osmosis. Then what do you mean by filtration? Filtration, when fluid is forcefully made to leave a compartment because of the difference in their hydrostatic and colloidal osmotic pressure. We call it as filtration. If the hydrostatic pressure in that compartment is more, it is favoring filtration. If the colloidal osmotic pressure in that particular compartment is more, it will oppose filtration. Then what is bulk flow? When large quantities of fluid is filtered, we are calling it as bulk flow. Then what is ultrafiltration? Filtration under high pressure is called as ultrafiltration. Then the last one in passive mechanism is solvent drag. What do we mean by solvent drag? When bulk flow is happening, when it is taking solutes along with it, then we call it as solvent drag. The solvent is dragging the solutes along with it. That is called as solvent drag. So that completes our discussion on passive transport mechanisms. Okay. Now we are moving on to the active transport mechanisms, right? So in active transport mechanisms, what do we have? We have primary active transport, secondary active transport. That is through the pumps. Occurring via pumps, we have the primary and secondary active transport. What do I mean by primary active transport? Sodium potassium ATPase pump is an example of primary active transport. Okay, so what is this pump doing? It is we already saw about sodium potassium ATPase pump, right? So I'm not going to again discuss in detail about it. It is basically creating a sodium gradient by actively pumping sodium from inside of the cell to the outside. Okay, to do this process, to pump 3 sodium out and 2 potassium inside, it requires energy. So it causes, with the help of the enzyme ATPase, it is causing breakdown of ATP into ADP and high energy phosphate is released. This energy is utilized for this transport mechanism. This is called as primary active transport. Now, these there is a gradient that is created by this pump. What gradient? Sodium gradient. So inside the sodium is less and outside sodium is more. So sodium will try to enter into the cell. Okay, when sodium is trying to enter into the cell, because of the gradient created by the sodium potassium ATPase pump, along with the sodium, glucose will also go. Okay, along with sodium, along with sodium, you can say glucose is also entering into the cell. Okay, with the help of a transport protein called as symporter. Okay, the name of the symport is called as SGLT, sodium glucose cotransporter. Sodium glucose co-transporter. It is a symport which is going to bring along with sodium, glucose is also coming into the cell. Such type of transport is called as secondary active transport. Why do we call it a secondary active transport? If you see here, you don't see any ATP here, but the energy that is released by hydrolysis of ATP in the primary active transport, this energy is utilized by this pump also. Indirectly, it is utilizing this energy for transport of glucose along with sodium into the cell. That is why such transport is called as secondary active transport. Okay, clear? So now let us look at different transporters. We have three different transport proteins. One is the uniport. Uniport is going to only transfer one ion in one direction. Sodium, potassium ions. Okay, the proteins that is going to transport it inside. That is called as uniport. Then we have symport. Just now we saw SGLT is a symporter. Example is symporter which is going to transport sodium and glucose into the cell. Basically symporter means it is going to transport two molecules in one direction, into the same direction. Then we have antiport. Best example is sodium potassium ATPase pump which is transporting sodium outside, potassium inside the cell. So basically it is called as antiporter. Okay, antiport means one molecule is entering into the cell, the other molecule is transferred out of the cell. That is called as antiporters. This is in general examples for sorry, various types of transport proteins. When we are talking about primary active transport, we have mentioned about the sodium potassium ATPase pump. The sodium potassium ATPase pump is also called as P-type pump. Okay. Then we have another one called as V-type pump. That is vacuole. Okay. Then we have one more called as F-type pump. Okay. Energy factor pump. Okay. Then we also have ABC transporters. Okay, so P-type pump or phosphorylation type pump, we have vacuole or V-type pump, we have F-type pump and ABC transporters. Example for ABC transporters are the cystic fibrosis transmembrane receptor protein, CFTR, and multi-drug resistance protein 1, 2, all these are examples for ABC transporters. F-type pump are the one which is present in a mitochondrial ATPase pump. Mitochondrial ATPase pump is an example for F-type pump which is present in the inner mitochondrial membrane. V-type pump or vacuole pump and this P-type pump, both these pumps are present in the cell membrane or the plasma membrane, and this F-type example for F-type pump is the hydrogen ATPase. Okay, clear all of you? Other examples for P-type pump, one example is sodium potassium ATPase. Okay, then hydrogen potassium ATPase is also an example for P-type pump. Okay, I think this is clear. So what we just now discussed is different types of active transport is not always only sodium potassium ATPase pump. We have different types of pump. They all come under different categories and they are specifically located at different locations. That is what I wanted to tell you. ABC transporters are also located in the cell membrane. Okay. So with this, let us move on to the vesicular transport. Vesicular transport, as the name indicates, it is going to happen by formation of vesicles. So far we saw active and passive pump along with the need along with ATP, and it does not only requires ATP but also concentration gradient. Active pump against the concentration gradient, passive pump along the concentration gradient, right? Now this vesicular transport is an example of active transport, but doesn't depend upon the concentration gradient. Okay, it is a active transport that utilizes metabolic energy, but concentration gradient is not important here. Vesicular transport, how does it occur? By either formation of the vesicle or fusion of the vesicle. If it happens by fusion of the vesicle, fusion of vesicles to cell membrane. We call it as exocytosis. Example, release of neurotransmitter, release of hormones, all happen in this fashion through exocytosis. Okay. Now, fusion, that is exocytosis. Formation of vesicle. Formation of vesicles. If the transport mechanism happens by formation of a vesicle, then we call it as endocytosis. Endocytosis of all these are very, very important MCQ terms. Fusion of vesicle. Example for fusion of vesicle is what type of transport. Formation of vesicle is what type of transport. So now endocytosis is formation of vesicles, right? So three endocytosis again of three types: phagocytosis, pinocytosis, receptor-mediated endocytosis. Okay, so what do we mean by phagocytosis? When the cell is engulfing large macromolecular substances from the extracellular matrix into the inside of the cell by formation of vesicle, we call it as phagocytosis. Okay, pinocytosis is also called as cell drinking. Phagocytosis is called as cell eating. Pinocytosis is called as cell drinking. That is, pinocytosis is the fluid portion of phagocytosis. So when the cell is forming a vacuole or a vesicle, large fluid component of the extracellular fluid along with the particular substance that needs to be transported will be brought into the cell. So this when the substance is transported into the cell, even the fluid is also coming along with it. Extracellular fluid is also coming along with this. This is called as pinocytosis. And then we have another one called as receptor mediated endocytosis. What is this receptor mediated endocytosis? In the cell membrane we have specialized region called as coated pits. Okay, in these coated pits you can see specific receptors are present in these coated pits. Okay, so these receptors, the particular substance that is going to enter into the cell, the molecule that is going to be transported into the cell will come and attached with these receptors. Okay, once they are attached to the receptors in this region of coated pits, then a vesicle will be formed and the substances will enter into the cell. Such type of transport is called as receptor mediated endocytosis. Okay, and transport across epithelia we already saw, transcellular route and paracellular route. So that finishes our discussion on transport, various types of transport mechanism across the cell membrane. Okay. Okay. Now let us move on to the next question. A 55-year-old patient with rheumatoid arthritis. Okay, we have a 55-year-old patient with rheumatoid arthritis who is being treated with a non-steroidal anti-inflammatory drug. So the disease here is rheumatoid arthritis. It is a chronic inflammatory condition. Okay, so since it is a chronic inflammatory condition, they are treating the patient with non-steroidal anti-inflammatory drug. Despite some improvement, the patient continues to experience joint inflammation. So, patient has rheumatoid arthritis. It is a chronic inflammatory condition. And for this inflammatory condition, they are giving a non-steroidal anti-inflammatory drug. Despite treatment, the patient's symptom persists. That is, the inflammation is persisting, because of which the patient is having pain. Now they are asking which of the following inflammatory mediator is most directly responsible for chronic inflammation seen in rheumatoid arthritis and whose activity is not significantly affected by NSAIDs. That is, they are, two questions are being asked in this. One is, which inflammatory mediator is responsible for inflammation and pain in rheumatoid arthritis? That is one. Second thing, which inflammatory marker non-steroidal anti-inflammatory drugs will have no effect? So both the questions are being asked here. They are given you four options: tumor necrosis factor alpha, interleukin 6, prostaglandin E2, leukotriene B4. All these four are markers of inflammation. Okay, so among these four markers of inflammation, you have to select one which is responsible for the inflammation in rheumatoid arthritis and which non-steroidal anti-inflammatory drugs will not be able to act upon. So that is going to be your answer. So for this, first we should know about rheumatoid arthritis a little bit. So as I already mentioned, it is a chronic inflammatory condition in which pro-inflammatory cytokines like CNF alpha and interleukins play a role. Okay, so in this option we have both of these things, tumor necrosis factor alpha as well as interleukin. Both are cytokines. Okay, both are cytokine and both of them play a role in inflammation in what do you say, rheumatoid arthritis. Okay, then what about prostaglandins and leukotriene? Both prostaglandins and leukotrienes are eicosanoids. Okay, so these are eicosanoids. What are they going to do? Prostaglandin is formed from arachidonic acid via the cyclooxygenase pathway, COX pathway. Okay, whereas leukotriene is also formed from arachidonic acid via the lipooxygenase pathway. Okay, now we all know non-steroidal anti-inflammatory drugs bring about their mechanism of action acting via the inhibiting cyclooxygenase enzyme. Okay, just now I told you in rheumatoid arthritis, the main mediator or inflammation is cytokines, tumor necrosis factor alpha and interleukins. Okay, that is why in this patient, the non-steroidal anti-inflammatory drug is not able to bring about its action because it is going to act on cyclooxygenase and reduce the production of prostaglandin, not interleukins and tumor necrosis factor alpha. Okay, so the answer here, you have to select between tumor necrosis factor alpha and interleukin 6. Among these two, tumor necrosis factor alpha is the most important pro-inflammatory cytokine. So I will go with TNF alpha. So non-steroidal anti-inflammatory drug is not effective against TNF alpha because this is a cytokine, and non-steroidal anti-inflammatory drugs are going to act on eicosanoids, especially prostaglandins, by inhibiting the enzyme cyclooxygenase. Okay. So that is what is written here. NSAID primarily targets the cyclooxygenase enzyme to reduce the production of prostaglandins. And prostaglandins are mainly involved in mediating fever and pain during inflammation. Okay, they do not significantly affect the cytokine production. And the key mediator of inflammation in rheumatoid arthritis is TNF alpha. Okay. So let us also look at the sources and their role in inflammation. So just now we saw TNF alpha, we saw, okay, it promotes inflammation and joint damage. This is a cytokine which is activated by macrophages and T cells. Similar to tumor necrosis factor alpha, interleukin 6 is also activated by macrophages and T cells, and it is an stimulates the acute phase response during inflammation. Prostaglandin E2 and leukotriene B4, they are produced from arachidonic acid via cyclooxygenase and lipoxygenase enzymes. Okay, and mainly present in the leukocytes and macrophages. They mediate pain, fever, and inflammation. Additionally, leukotriene also attracts neutrophils and promotes inflammation. Okay, so now I think you're clear with that question. Now we'll move on to the next question, which is also in similar lines. Which of the following inflammatory mediators is primarily responsible for inducing fever during an inflammatory response? Just now we saw what was the inflammatory mediator. Here only we saw. Prostaglandin is an inflammatory mediator which mediates pain, fever, and inflammation. Okay. So among the given four options, which is the right answer? Which of these inflammator mainly mediates pain and fever? It is prostaglandin. Okay. Then what about histamine, leukotriene, and cytokines? Cytokines we saw. What are those cytokines? TNF alpha, interleukin 1, interleukin 6, etc. Okay. All these are pro-inflammatory cytokines which is mainly going to cause inflammation. Whereas fever and pain will mainly be brought about by prostaglandins. Cytokines will also bring about same effects not directly, by indirectly stimulating the production of prostaglandins. Okay, so that is why cytokines is not the answer here. What about histamine and leukotrienes? Okay, histamine is very important in allergic phenomenon. Okay, it will also cause vascular permeability. It will increase the increasing the vascular permeability. Okay, it is also causing vasodilation. Rather than fever and pain, these are the important effects of histamine. Histamine is also important in gastrin secretion. Okay, so histamine has a role in all these areas. What about leukotrienes? Leukotrienes are also very important for chemotaxis. Okay, it is important for chemotaxis. And leukotriene is important, it causes bronchoconstriction. So it has a role in asthma and allergy. Rather than pain and fever, if you see leukotrienes, these are the function: chemotaxis, bronchoconstriction, role in asthma, allergy. Even leukotriene increases the vascular permeability, vasodilation. These are effects of leukotriene. Histamine, again the same thing: increase vascular permeability, vasodilation, gastrin secretion. All these are roles of histamine. Prostaglandin, when you see the word pain and fever, it corresponds to prostaglandin. And how does prostaglandin bring about fever? It acts on the hypothalamus, altering the physiological set point for temperature. Okay, they act on the hypothalamus to raise the body set point for temperature, leading to fever. Okay. Now, coming to the next question. Which of the following statements regarding homeostasis and its mechanism are true? So, next we are moving on to homeostasis. Okay, so what do you mean by homeostasis? So, first let us look at the definition. Before looking at the options, let us look at the definition of homeostasis. What do we mean by homeostasis? Homeostasis is defined as the maintenance of constant internal environment. Maintenance of the constant internal environment of the body is called as homeostasis. How does body maintain its homeostasis? By two regulatory mechanisms: negative feedback mechanism and positive feedback mechanism. What do we mean by negative feedback mechanism? It is a mechanism in which it is going to trigger processes which will either inhibit or enhance the formation of substances, so that the altered variable is going to come back to its original physiological set point. Okay, so here you can see. So this is an example of negative feedback mechanism. Negative feedback mechanism can operate in both the direction. Suppose a particular variable has been altered from its physiological set point. Okay, if now we know our mean arterial pressure is around 95 to 100 mmHg. Okay, so now this is the physiological set point for mean arterial pressure. If this pressure increases, okay, if it increases to 150, then what will happen? It will act in the opposite direction. It will reduce the blood pressure and bring it to the physiological set point. Suppose the patient is having hypotension. This pressure has gone to 60. Then what it will do? It will act again in the opposite direction. It will raise the blood pressure. Now, when the pressure is in 60, it is going to activate processes or mechanism which is going to increase the blood pressure and bring it back to its 95. If the pressure is high, what is it going to do? It is going to activate mechanism which is going to decrease the blood pressure. So, negative feedback mechanism can act in either direction. Okay, and negative feedback mechanism is the most important mechanism for establishing homeostasis. Okay, then we have positive feedback mechanism. Positive feedback mechanism, what is it going to do? It is a feedback mechanism in which the variable is going to trigger processes that is going to further enhance the production of the variable. Okay, suppose a blood clot is formed. Example of blood clotting. Okay, activation of few platelets is going to activate more platelets until a temporary hemostatic plug is formed. So there are many examples of positive feedback mechanism. Basically, positive feedback mechanism, it increases the change. It keeps on increasing the change. Until when it will be increasing like this? Until the physiological limit for that particular action is reached or the stimuli is cut off. Until then, it is going to keep on happening. So this is a vicious cycle. When you see the word vicious cycle, it always points towards a positive feedback mechanism. When you see the word maintenance of homeostasis, generally 99% of processes in the human body to maintain homeostasis is via the negative feedback mechanism. Okay. Now, positive feedback mechanism is either self-terminated or the trigger is removed. Important examples of positive feedback mechanism. Parturition reflex initiated by oxytocin. LH surge during ovulation. Hodgkin cycle, that is opening up of few sodium ions is going to cause opening up of more and more sodium ions, leading to massive influx of sodium and depolarization. So that is one sodium ion is going to open 10 more, the 10 more is going to open 100 more. So this is a positive feedback mechanism. Example, Hodgkin cycle. Okay. And also activation of the coagulation cascade, gastric enzymes like pepsin, all these are examples of positive feedback mechanism. Okay. Now let us look at the options. So what are they asking here? Which of the following statement regarding homeostasis and its mechanism are true? They are asking us the correct answer. Identify the correct answer. Homeostasis involves the regulation of internal conditions such as temperature and pH to maintain a stable internal environment. Yes, this is true. Okay, let us look at the other options also. Negative feedback mechanisms amplify changes in physiological process to restore equilibrium. Does it always amplify? No. Which is the one which always amplifies the signal? It is the positive feedback mechanism, not the negative feedback mechanism. So this statement is false. Then, hypothalamus plays a critical role in maintaining the body temperature by initiating responses such as sweating or shivering. Yes, this is true. Hypothalamus is the seat of regulation for body temperature, for food intake. Many functions are there. So regulation of body temperature is one very important function of hypothalamus. Correct. Positive feedback mechanisms are the primary way the body maintains stable internal conditions during stress and injury. No, this is false. So now here they have asked us to identify the true statement. We have two true statements here. Then which one will we choose? Will we choose the role of hypothalamus or temperature and pH? Obviously, we will have to choose the option A because it is more important compared to the role of hypothalamus. Both are true, but when it comes to pertaining to homeostasis, option A seems more appropriate compared to option C. Okay, so the right answer is option A. Example of negative feedback mechanism, most of the hormones are regulated via secretion of hormones are regulated via the negative feedback mechanisms. Okay. The next question. We have a 62-year-old diabetic patient. 62-year-old diabetic patient presents with frequent episodes of hyperglycemia. So we have a diabetic with hyperglycemia. That means uncontrolled diabetes. Despite treatment with insulin, the patient is on insulin. Still, he is having hyperglycemia. Laboratory tests reveal elevated blood glucose levels. Obviously, when there is frequent episodes of hyperglycemia, blood glucose will be elevated and presence of ketones in the urine. This condition suggests an impaired glucose regulation. See, in all these questions, they are already telling you what is getting impaired. What they are asking is because of this impairment, what physiological process is getting affected, or alteration in which physiological process has led to this particular impairment. That is what the question, most of the questions are there. So now we have a scenario where glucose homeostasis is altered, leading to hyperglycemia. Now what are they asking? Which of the following primary homeostatic mechanism is compromised in this patient? So they have given us four options, and among these four options we have to select which physiological function has been altered which is resulting in hyperglycemia in spite of insulin being present there. Okay. Option A is gluconeogenesis regulation in the liver. What do we mean by gluconeogenesis? Gluconeogenesis means production of carbohydrate from production of energy from non-carbohydrate sources. Okay. Is this the right answer? Because of this only the blood glucose level is high and the patient has uncontrolled diabetes mellitus, or insulin receptor sensitivity in the target tissue is compromised, or the glycogen synthesis in the liver is compromised, or renal glucose reabsorption is compromised. So among these four options, which do you think is the right answer? Yes, obviously it is the insulin receptor sensitivity in the target tissue that is being compromised. Because of that only, in spite of insulin being present, the patient's cells are not taking up the glucose, resulting in hyperglycemia. Okay, so what is happening? Look at this picture. Okay, so here in this picture you can see the mechanism of action of insulin. Okay, if I have to summarize the function of insulin in one line, what will we say? Insulin causes peripheral utilization of glucose. What do we mean by this word peripheral utilization of glucose? Peripheral utilization of glucose means it is going to cause, or it is going to lead to entry of glucose into the cell. Okay, so when insulin is there, glucose has to enter into the cell. When glucose enters into the cell, what will happen? The blood glucose levels will come down. This is a normal physiology. In this patient, what has happened? He is a diabetic and on insulin. So we are giving insulin exogenously. Even after giving insulin, the glucose has not entered into the cell. Why? For that, we need to understand this mechanism of action of insulin. So this is insulin. Okay. Insulin is a polypeptide hormone. So the receptor for insulin is going to be present on the cell membrane. Okay, a cell surface receptor is there. So this is the insulin receptor. So we have the insulin receptor attached on the cell membrane. When this insulin comes and binds to the receptor, forming hormone receptor complex. When hormone receptor complex is formed, then only phosphatidyl inositol 3 kinase will get activated. Once this gets activated, what is it going to do? It is going to cause the fusion of the endosome that contains glucose transporter, to the cell membrane. Okay, glucose transporter to the cell membrane. Why this glucose transporter from the interior of the cell has to come and fuse on the cell membrane? Only if this glucose transporter is fused to the cell membrane, what will happen? Glucose will enter into the cell. Glucose will enter into the cell, right? Facilitated diffusion of glucose will happen. From the region in blood, the glucose will be high. Glucose transporters are going to transport glucose from outside into the cell. This is an example of facilitated diffusion, right? So now this will only happen, all this will happen only when insulin binds with the insulin receptors and forms hormone receptor complex. Now when this receptor is not present, or this receptor number is reduced, or if the receptor is not functioning properly, we call that condition as insulin resistance. Okay, insulin is present, but the receptor is defective. So the insulin is not able to act and form insulin hormone receptor complex. So if hormone receptor complex is not formed, what will happen? Peripheral utilization of glucose will not happen. So more glucose will be present in the bloodstream, leading to hyperglycemia. That is what is happening to our patient. Okay. So now when glucose is not utilized for energy production, lipolysis is happening. Ketone bodies are getting formed in this patient. Okay. Now is it clear? Why the answer is insulin receptor sensitivity in the target tissue? That is why the right answer is insulin receptor sensitivity in the target tissue is compromised, because of which only this patient is having hyperglycemia despite availability of insulin. Okay. Next. Which of the following factors does not typically influence the basal metabolic rate? Does not influence basal metabolic rate. They have given 4 factors here: age, body composition, physical activity level, hormonal status. Among these 4 options, which does not affect the basal metabolic rate? For that, first we should know what is basal metabolic rate. What is BMR or basal metabolic rate? It is the body's ability to expend energy when the body is at rest to carry out normal physiological processes. Okay, it is a rate at which the body expends energy at rest to maintain vital physiological function. So this is defined as BMR. BMR depends upon the age. Yes. Why it depends upon the age? Because as we age, we lose muscle mass. When we lose muscle mass, the basal metabolic rate reduces. So definitely age influences BMR. What about body composition? Same thing. If you have more muscle mass, better will be the basal metabolic rate of the person. So body mass composition also has a role when it comes to BMR. What about physical activity level? Physical activity level or physical exercise, physical activity level is going to affect the total energy expenditure. Okay, the amount of calorie that the person is burning. The total energy expenditure is only affected by physical activity level. Physical activity level does not affect the BMR directly. Okay, and then let's look at the hormonal status. Hormonal status also affects the BMR. Okay, especially which hormone? Thyroid hormone. Okay, so thyroid hormone increases BMR. Body composition also affects BMR. Age also affects BMR. So the right answer here is physical activity level. Let's go for a short break and we'll be back soon. So welcome back. Now let us look at the next question. Which of the following statements regarding nitric oxide and its physiological roles are true? So they are asking a direct question about nitric oxide and its physiological functions. So as usual, before answering this question, let us look at the functions of nitric oxide. Okay. Nitric oxide is a very, very important critical signaling molecule. It is a gas. Okay. It is formed along with oxygen as a cofactor from L-arginine. L-arginine in the presence of oxygen as a cofactor with the help of the enzyme nitric oxide synthase forms nitric oxide. So where is the nitric? So first we saw about the synthesis of nitric oxide. Okay, nitric oxide is synthesized from L-arginine with the help of the enzyme nitric oxide synthase in the presence of oxygen. Okay, is it clear? This is one very important MCQ. Now, coming to the functions of nitric oxide. Okay, I already told you it is a very, very important signaling molecule. It is a gaseous signaling molecule. What are its functions? Here I have listed out some very important functions of sorry, nitric oxide. The first and the most important function of nitric oxide is vasodilation. Okay, in cells that have endothelial dysfunction, vasodilation is absent or vasodilation is diminished. Why? Because nitric oxide is not formed properly. Sorry. Since nitric oxide is not formed properly in cells with endothelial dysfunction, vasodilation is not proper. So nitric oxide is causing relaxation of blood vessels, thus it decreases blood pressure. That is one very important function. Then the next function is it helps a role in neurotransmission. It helps in communicating between the neurons. It is also involved in processes like learning and memory. When it comes to immune response, nitric oxide plays a very important role in the immune system by helping to kill pathogens and it modulates inflammation. Macrophages, for this to happen, macrophages produce nitric oxide which invades the microorganisms. Then for cellular signaling, it acts as a second messenger for various pathways. Okay, thus bringing about apoptosis, cellular growth and gene expression. It regulates platelet function. It inhibits the platelet aggression, prevents excessive clotting and promotes healthy blood flow. It not only causes the relaxation of blood vessels, nitric oxide also causes relaxation of smooth muscle. Mainly helps in digestive process. Okay, so now that we saw the functions of nitric oxide, let us look at the options and find which is the right option here. What do they say? In the first option, they are telling us nitric oxide is a potent vasodilator that helps to regulate blood pressure and improve blood flow. Is this true? Yes, this is true. Then nitric oxide is synthesized from arginine by the enzyme nitric oxide synthase and requires oxygen as cofactor. Yes, this is also true. Nitric oxide functions as a neurotransmitter in the central nervous system, playing a role in memory and learning. Yes, this is also true. Nitric oxide has a long half-life in biological systems and due to its stability makes it an ideal therapeutic agent for chronic condition. This is false. It has a very short half-life. Okay. So which is the answer here then? What have they asked? They have asked which has true. All the first three options are true. Ideally, this question has to be which among the following is false. Which among the statement is false? If they've asked like that, then we would have selected option D. Now we have three options which are true. From these three options, you have to select the best answer. If we have to select the single best answer from these three options, I would go with option A. It is a potent vasodilator that helps to regulate blood pressure and improve blood flow. Okay. Okay. Now going to the next question. Which type of aphasia is characterized by fluent but nonsensical speech. So underline the key terms. We are talking about aphasia. This aphasia is fluent and nonsensical speech. The patient has significant difficulty in comprehension. Okay. What do you mean by nonsensical speech? The person is he's talking something, but it totally incomprehensible. We are not able to understand. Okay, that is nonsensical speech. Second is difficulty in comprehension. That is what we are not able to understand it. So they have given four options: Broca's aphasia, Wernicke's aphasia, global aphasia, and anomic aphasia. So among these four, in which aphasia we are going to have fluent nonsensical speech, and the patient will also have difficulty in comprehension. That is, we will not be able to understand what this patient is talking, and neither the subject is able to comprehend what we are talking to them. Okay, so in which of this aphasia you're going to get such a scenario? So before finding the answer, let us look at aphasia, its types and causes. Okay, so first, what do you mean by aphasia? Aphasia is a communication disorder. Aphasia is a communication disorder. So because of this disorder, what is happening? A person's ability to process and produce language is affected. Very important. The ability of the person to process and to produce language is affected. That means to understand as well as to talk. Both is affected for in case of aphasia. So when a person is having aphasia, it can have an impact on his speaking, on his understanding, on his reading as well as writing. All these domains can be affected. Reading, writing, speaking and understanding. All the four domains, all these important aspects of a language can be affected. Okay. So now different types of aphasias are there. First, let us look at this picture. I will show you. It will be easier for you to understand and remember. Okay. So when it comes to aphasia, already I told you, communication disorder. Broadly, we can divide it into receptive and expressive. Receptive means what? Understanding. Expressive means what? Expressing, speaking. Right? Now we have certain areas which is important for language. What are those areas? We have Broca's area, Wernicke's area, angular gyrus and arcuate fasciculus. So Wernicke's area is nothing but area 22. Broca's area is area 44 and 45. Angular gyrus is area 39. This Wernicke's area and angular gyrus. Wernicke's area and angular gyrus are receptive. Receptive zones of language. Okay. Wernicke's area is important for comprehension. Comprehension. Okay. Angular gyrus is important for words. Okay, these are the key terms I want you to remember. When it comes to language, four important areas are there. Broca's area, actually receptive zones and expressive zone. Receptive zone means for understanding. So in the receptive zone, we have two important areas. One is Wernicke's area, the other one is angular gyrus. Angular Wernicke's area is area number 22, angular gyrus is area 39. Wernicke's area is for understanding or comprehension of the spoken words. I'm talking something, you are hearing it, you are processing and understanding what I'm communicating to you. Why? Because your Wernicke's area is completely normal, perfectly normal. Okay, something is written on at the same time I'm writing something in this slide. You are able to see, you are reading it and you are able to understand it. Why? Because your angular gyrus is also working in perfect condition. Angular gyrus, Wernicke's area, both are Examples of receptive zone, they are important for receptive language. And angular gyrus is for comprehension of written words. Wernicke's area is for comprehension of spoken words. Okay, well, we are left with what? Broca's area 44 and 45. This Broca's area, okay, so Wernicke, another name for Wernicke's area is sensory speech area. Okay, they can ask you any of these terms. That is why I am also telling the different terms that is there. Wernicke's area is called a sensory speech area. Sensory means comprehension, that's all. Broca's area is called as motor speech area. Broca's area is called as motor speech area. Okay, so what is Broca's area going to do? Expressive. So this is expressive. You are able to express. I am able to speak out because my Broca's area is working completely normal. Okay, so this is called as Broca's speech area. So the Broca's speech area is going to control the peripheral speech apparatus, the mouth, the larynx, the voice box. All these are controlled by the Broca's area, so that I am able to vocalize and produce the sound that I desire to. That is a function of Broca's area. This is for vocalization, motor output of speech. Okay, then what about the written speech? We have something called as Exner's area. Okay, we have the Exner's area, which is for writing. Okay, I am able to write, express it via writing. For that you need the Exner's area. So Exner's area and Broca's area is for expressive language. Wernicke's area and angular gyrus is for receptive language. Then what is the function of this arcuate fasciculus? Arcuate fasciculus is a bundle of nerve fibers which takes the information from the Wernicke's area. This is the Wernicke's area. This is the Wernicke's area. From the Wernicke's area, the information goes to the Broca's area. Okay, via the arcuate fasciculus. Okay, so this is a connecting system. So this is in broad. Now let us look at the different types of aphasia. That is a normal physiology of language, where it is produced and what is the role of different areas. Now this, there is a disorder in this communication. What are the different types of aphasias that can present with, and what is its clinical presentation? So Broca's area, also called as the motor speech area. If Broca's area is affected, we are going to end up having something called as non-fluent aphasia. Non-fluent aphasia, because why? This is motor. The person is understanding, but they are not able to express it. So non-fluent aphasia. What are the characteristic features of this aphasia? Difficulty in speaking fluently. The speech is often limited in short phrases. So when they are describing this patient, since most of the questions are not direct questions, it is a clinical case scenario. The description of the cases, the description of the patient's signs and symptoms is very important. We have to concentrate on that. Only when we understand that, we will be able to bring out the diagnosis and the reason why, which area is affected, because of which this particular disorder has occurred. That is what we have to finally come to a conclusion in most of these clinical case scenarios pertaining to physiology. So here, when they are telling there is a patient who is having difficulty in speaking fluently, the speech is often limited in short phrases, which area is affected? Immediately, what will you select? Broca's area. Okay, in our patient, it is not Broca's. There they are telling the speech is fluent but nonsensical. Okay, then what area will be affected? Wernicke's area. Okay, in our case, it is actually the Wernicke's area that is affected. So the aphasia is sensory aphasia or fluent aphasia, or also known as Wernicke's aphasia. Okay, now let us continue with Broca's aphasia. What is the location? Damage to the Broca's area in the frontal lobe. So that is Broca's aphasia. Then let us look at Wernicke's aphasia. Wernicke's aphasia is also called as fluent aphasia. What is the characteristic features here? We just saw nonsensical speech with difficulty in understanding the spoken as well as the written language. Okay, patients may produce long sentences, but it will not have any meaning. What is the location? Wernicke's area located in the temporal lobe. Then global aphasia. Global means both Wernicke's area as well as Broca's area is affected. So there will be severe impairment of both expressive as well as receptive language. The individual may struggle to both speak or understand language at all. So both the areas will be affected. Then comes anomic aphasia. Anomic aphasia means the patient will have difficulty in finding words, particularly nouns and verbs. So when you have a description of a disorder where they are telling the patient is having difficulty in producing, speaking, in finding nouns and verbs, that means you have to select anomic aphasia. Okay, speech is generally fluent but with frequent pauses. The defect in case of anomic aphasia is usually in the angular gyrus. Then comes conduction aphasia. What is the characteristic features of conduction aphasia? Conduction means what? It's conducted from one area to the other. So now you can easily think what is the defect will be in the arcuate fasciculus, because arcuate fasciculus is the one which is connecting the Wernicke's area to the Broca's area. When this connecting arcuate fasciculus is damaged, then it results in conduction aphasia. What is the, now you know the name of the aphasia, but what is the clinical presentation of the subject with conduction aphasia? They will have difficulty in repeating phrases and making spontaneous speech errors. Comprehension is usually intact. The comprehension is usually normal, but they will not be able to repeat what we are telling. Okay, that is conduction aphasia. Now, broadly, these are the 5 different types of aphasia. Now, other important terms also they keep asking related to aphasia. What is it? Pure word blindness, pure word deafness, pure word mutism. This also you should know. What do we mean by pure word blindness? This is also called as auditory verbal agnosia. Okay, this is pure word deafness. Please make a change. Okay, what is pure word deafness? The pure word deafness, another term is auditory verbal agnosia. When you see the word auditory, it means deafness. Okay, pure word deafness, also known as auditory verbal agnosia, there is impairment of auditory comprehension and repetition and inability to write following dictation. If you check the auditory pathway for this patient having pure word deafness, if we check the hearing of this patient, the hearing is completely normal. But still, if you are giving a dictation, they will not be able to write it. Why? Because they are not able to understand. The hearing is normal, but they are not able to understand what you are dictating, so they will not be able to even write it. Okay, that is called as pure word deafness. The lesion is in superior temporal gyrus of both the sides. Okay, then comes pure word blindness. What do we mean by pure word blindness? Just the opposite of pure word deafness. This is also called as visual verbal agnosia. Okay, this is characterized by inability to read aloud, to understand the written script, and to name colors. The vision is completely normal, but still, if you give them, if you ask them to read from this PowerPoint, they will not be able to read. They are seeing something, you ask them to name the color, they will not be able to tell. Okay, so that is called as pure word blindness. Now, where is the lesion in case of pure word blindness? Left visual cortex, mainly near the geniculocalcarine tract. Then comes the last one, pure word mutism. What do you mean by pure word mutism? The characteristic features of the characteristic feature of pure word mutism is loss of capacity to speak while perfectly retaining the ability to write. Loss of capacity to speak while perfectly retaining the ability to write. That is why it is called as mutism. They are not speaking, but they are able to write. They are able to understand what is spoken to them. They are able to read silently. They are not able to produce the sound. They are able to read silently with comprehension and repeat the spoken words. This is called as pure word mutism or aphinmia. Location is a lesion which is located in the dominant frontal lobe. mainly either due to a vascular lesion or a localized injury to the frontal lobe of the dominant side. For a right-handed person, it is a left, and left-handed person, it is a right. Dominant lobe, if the frontal lobe is on the dominant side, when the frontal lobe is damaged, it will lead to pure word mutism. Okay, so with that, I think we have covered almost all the questions that will be coming from aphasia. You will be able to answer most of the questions from aphasia. Okay, now what are the causes of aphasia? Most important common causes: disruption of blood flow to the brain, leading to because of stroke. Then when there is a traumatic brain injury, brain tumors damaging or putting pressure on the language processing area. Neurodegenerative disorders like Alzheimer's disease or frontotemporal dementia, all this will cause progressive language impairment. Whenever you have a history of neurodegenerative disease, there will be always progressive language. With all of a sudden, in one day, they will not lose their ability to communicate. It will be progressively progressive loss of language. Infectious conditions like encephalitis and also certain types of seizures can also affect the language areas. Okay. Now, next question. We have a 47-year-old man who reports excessive daytime sleepiness, EDS, excessive daytime sleepiness, and difficulty maintaining sleep at night. Despite having a seemingly adequate amount of sleep, he often feels unrefreshed upon waking up. All these are classically symptoms of sleep disorder breathing. Okay, SDB, sleep disordered breathing. Different types of sleep disordered breathing are there. The clinical scenario which is being shown here is mainly pointing towards sleep apnea. Okay, so let us see what else are they giving. Okay, a polysomnography reveals intermittent cessation of breathing during sleep. So now polysomnography is the definitive test to confirm obstructive sleep apnea. So in polysomnography, intermittent cessation of breathing, basically what they are telling, apneic episodes are happening. Which of the following sleep disorder is most likely cause of this symptom? So we know it is a direct answer. It is obstructive sleep apnea, OSA, or simply sleep apnea, any of this option. So they've given 4 options here: insomnia, obstructive sleep apnea, narcolepsy, restless leg syndrome. So what is the answer here? The answer is obstructive sleep apnea. Now let us see a little bit about obstructive sleep apnea. When you have the description of the patient, sleep apnea is a disease which can be confirmed with the clinical signs and symptoms of the patient itself. Okay. Sorry. So what are the classical presentation of a patient with obstructive sleep apnea? Obesity, excessive daytime sleepiness, very cardinal feature of OSA. Because of the unrefreshing sleep at night, because of the multiple apneic episodes that happens during the night, the patient will have micro arousals. Because of that, even if they are sleeping for 8 hours, the next day morning when they wake up, they have an unrefreshed feeling. Excessive daytime sleepiness, and polysomnography will show an apnea hypopnea index of more than 5. So here they've directly given intermittent cessation of breathing, that is apneic episodes are happening. Usually AHI, apneic hypopneic index of greater than 5 is diagnostic of obstructive sleep apnea. And then what is this polysomnography? Polysomnography is also called as sleep study. Polysomnography is also called as sleep study. Then what is an hypnogram? Hypnogram. Hypnogram, you would have seen something like this, no? So what is this? This is a hypnogram. Hypnogram is showing you the sleep cycle. Hypnogram only shows you the sleep cycle, that is different stages of sleep. Different stages of sleep is shown in a hypnogram. Then what is a polysomnography? Polysomnography is also called a sleep study. In polysomnography, along with the EEG, various other physiological parameters will also be recorded simultaneously over the entire night. Okay, and if they suspect obstructive sleep apnea, or if they have diagnosed obstructive sleep apnea in polysomnography, they will even do a CPAP titration in the same polysomnography setup. So that is called a sleep study. Sorry. So what are all the physiological parameters that is recorded in a overnight sleep study or polysomnography? Most important is the EEG, that is the electroencephalogram. Along with the EEG, we will also record EOG, that is electrooculogram, EMG, activity of the muscle. We will check for oxygen saturation, snoring. Okay, another very important classical symptom in a patient of obstructive sleep apnea is snoring, history of snoring at night or history of snoring during sleep. Then chest and abdominal movements, ECG, okay, sometimes even blood pressure, nasal airflow. All these parameters will be seen in polysomnography. Okay, polysomnography is not merely an EEG. In polysomnography during sleep, so what is polysomnography? It is a a technique in which during sleep, along with EEG, various other physiological parameters will be recorded simultaneously. That is called as polysomnography. Okay, so I think that makes you clear about OSA. And in polysomnography, an apnea hypopnea index of greater than 5 is indicative of obstructive sleep apnea. Okay, so that is with polysomnography, hypnogram, and sleep apnea. Let us, since they have given other disorders like insomnia, narcolepsy, restless leg syndrome, all these are sleep disorders. Let us look a little bit into these disorders as well. What do we mean by insomnia? What do we mean by insomnia? Insomnia is nothing but a chronic ability to sleep in spite of adequate opportunity to do so. Okay, chronic inability to sleep. The patient will not be able to sleep, or to put it in simple words, the patient is not able to sleep. Inability to sleep is called as insomnia. Okay, then what is narcolepsy? This is a condition in which there is episodic sudden loss of muscle tone accompanied by irresistible urge to sleep. Muscle tone is completely gone and the patient wants to sleep. Irresistible urge to sleep. Okay, so since the muscle tone is completely lost here, immediately you can correlate and say that a patient of narcolepsy will start sleeping with REM. Okay, normally for others, normal person, the sleep will progress from awake, drowsiness, stage 1, 2, 3 of NREM sleep, then again stage 3, 2, 1, then only REM sleep will come. But in a person of narcolepsy, they will directly sleep in REM phase, okay, which is very characteristic. So when we do a sleep study, we will see REM sleep will, the patient with narcolepsy will have no sleep latency, and also immediately they will fall into the REM phase of sleep. Okay. And also the hypothalamus of narcoleptic subjects contain less hypocretin producing neurons. Then what is cataplexy? Cataplexy is a condition in which there is sudden loss of power of movement and posture while the subject is awake. Okay, followed by irresistible desire to sleep. Then what is bruxism? Bruxism is nothing but teeth grinding, usually seen in children, associated with dreams and REM sleep. Okay, then what is nocturnal enuresis? Bedwetting at night in children above 8 years of old. When does nocturnal enuresis become pathological? In children even beyond 5 to 8 years, if they still continue bedwetting, then only we call it as pathological. But for us, from physiology point of view, nocturnal enuresis means what? Bedwetting in the night. Okay. When we do a EEG, it is usually seen just before the just before an episode of bedwetting. A burst of rhythmic delta waves is seen in the EEG, and also there is generalized body movements. Then what is nocturnal paroxysmal dystonia? This is nothing but choreoathetotic or ballistic movements are occurring during NREM sleep. So that is called as nocturnal paroxysmal dystonia. Then sleep paralysis. Sleep paralysis is nothing, most of us would have experienced the sleep paralysis. It is normal. It usually occurs during the transition from sleeping to waking up state. During this phase, the patient is unable to activate their muscle, but they are awake, but they're unable, they feel completely paralyzed. Okay, it occurs towards the end of REM sleep. Then two important things: night terrors and nightmares. Night terrors usually is a childhood disorder, occurs during NREM sleep. These are the key words I wanted to remember. That is why I didn't write much. Two words you have to remember. And night terror means it occurs in it's a childhood disorder. REM sleep disorder, NREM sleep disorder, slow wave sleep disorder. Nightmare. What do you mean by a nightmare? This is nightmares are more common in adults. It is a REM sleep disorder. Nightmares are REM sleep disorder in adults. Night terror is a childhood disorder. It occurs in children and it is an NREM sleep disorder. Okay. Then sleepwalking or somnambulism. Most commonly, somnambulism means sleepwalking. It most commonly occurs during NREM sleep, that is during the slow wave sleep or deep sleep, and patient is not aware of it on waking up. Okay, so that completes our discussion on various sleep disorders. Let's move on to the next question. We have a 45-year-old female with history of hypertension and diabetes. So she has both metabolic diseases, hypertension as well as diabetes. Now she is presenting with symptoms of headache, confusion, sudden onset of left-sided weakness. So the brain is involved. Headache is there, confusion is there, left-sided weakness is there. A neuroimaging reveals a contrast-enhancing lesion in the brain. Whenever you see a contrast-enhancing lesion in the brain, we all know the brain is protected by the blood-brain barrier. Okay. So this blood-brain barrier is going to limit the substances that is going to enter into the brain capillaries, into the blood. But now, whenever there is a lesion like this, whenever there is a tumor in the brain, or there is an inflammatory condition, there is an infection, some pathology that is going on in the brain tissue, in all these areas, the blood-brain barrier will be compromised. When in the region of the tumor or in the region where an inflammation is going on, there is an infection, all this will cause disruption of the blood-brain barrier. So once in these region, when the blood-brain barrier is disrupted, what is going to happen? You are giving a contrast. The dye will be taken up by that area that is disrupted. Easily the contrast is going to enter there. Because of the disruption of the blood-brain barrier, the contrast will be taken up by the structure. That is why they do a neuroimaging. When they in the neuroimaging, they see a contrast enhancing lesion, means the contrast is taken up by that lesion. Why? Because the blood-brain barrier is damaged. Look at the next line. Given the involvement of blood-brain barrier in this case. Now do you understand why blood-brain barrier is involved in this case? Okay. Given the involvement of blood-brain barrier in this case, which of the following statement best describes the role of blood-brain barrier in context to brain pathology? So basically they're asking the function of blood-brain barrier. You have to identify that. So let us first, as always, revise our basics. Let us see what is this blood-brain barrier, what is it made up of, what are the substances that is going to enter, and what are the substances that can leave through the blood-brain barrier, and what are the functions of blood-brain barrier. Okay, so blood-brain barrier is mainly formed by tight junctions of the endothelial cells and food processes of the astrocytes. So what are the structures that forms the tight junction? Endothelial cells of the capillaries and food processes of the astrocytes forms this tight blood-brain barrier. There are certain areas in the brain which is naturally devoid of this blood-brain barrier, which normally do not have a blood-brain barrier. Those structures are together collectively called as circumventricular organs. So what are circumventricular organs? Those are regions in the brain which is devoid or which does not have a blood-brain barrier. So what are those areas? The median eminence of hypothalamus, the subpharyngeal organ, OVLT, organovasculosum of lamina terminalis, area postrema, that is the chemoreceptor trigger zone, and then posterior pituitary, that is a neurohypophysis. All these areas, 1, 2, 3, 4, 5. So all these 5 areas are regions, very important for you to have to memorize and remember this, because these are called as circumventricular organs. These are regions where blood-brain barrier is absent. Okay. Now, what is this blood-brain barrier? As we just now saw, it's a selectively permeable barrier which allows only certain substances to enter into the CNS. By restricting the entry of substances, it is basically protecting the brain from harmful toxins and pathogens. Okay. At the same time, it allows essential nutrients to pass through it. Now, what are all the substances to which it is highly permeable, to which it is impermeable, and it is moderately permeable? That is also an important MCQ. You see here, highly permeable to water, any lipid soluble substances like gases and lipophilic substances like free forms of steroid hormone, alcohol, and anesthetic agents. So, it is highly permeable to water, gases, and lipid soluble substances. Slightly permeable to electrolytes like sodium, potassium chloride. It is slightly permeable to electrolytes. Totally impermeable to plasma proteins and protein bound forms of steroid hormones, protein antibodies, and most non-lipid soluble large organic molecules. So basically all proteins and polypeptides cannot enter the blood-brain barrier. Lipid soluble substances, gases, and water can easily enter the blood-brain barrier. Important vital nutrients and electrolytes have moderate permeability. Okay. Now, as I already told you, in pathological conditions, the blood-brain barrier is compromised. What are the functions of the blood-brain barrier? One is protects the brain by selectively allowing certain substances, and at the same time it also prevents the escape of neurotransmitter and maintains the ionic constancy in the brain fluids. So that is with the structure function of blood-brain barrier. Now let us look at the options. What they are asking is, among these 4 options, which of the following statement best describes the role of blood-brain barrier in the given subject? Okay. So the blood-brain barrier prevents all therapeutic drugs from entering the central nervous system. Is it true? No. The blood-brain barrier is primarily composed of astrocytic end feet that restricts drug diffusion. But it also involves the not only astrocytes but also the endothelial cells of the capillary. So even that is not the right option for the given pathology. The blood-brain barrier can be disrupted by inflammation, allowing larger molecules and contrast agents to cross. Yes, this is the answer. Let us look at the last option. The blood-brain barrier exclusively regulates the passage of ions and does not affect the permeability of larger molecules. No. Okay. So what is the right answer? Option C. Okay. Now we have another question. A 53-year-old patient presents with difficulty. A 52-year-old patient presents with difficulty in planning and organizing tasks. So it's a CNS disorder. The patient has problem with planning and organizing tasks. So planning, organizing tasks, these are all called as executive functions. Okay, these are executive functions. So the executive function is lost. Alongside, the patient is also having personality and social behavior issues also. Neuroimaging reveals atrophy in the frontal and temporal lobes. Okay, executive function is actually the function of the frontal and temporal lobes. Based on these findings, which of the following cognitive function is most likely impaired in this patient? Which is impaired here? I already told you. The executive function is impaired. Okay. They have given other options as well. After finishing this question, we will take a short break. So the other function, let us look at the other options as well. First option is visual spatial processing. The second is executive function, that is what is lost in this patient. Then we have memory retrieval and then language comprehension. We'll just see which area is responsible for all these functions, and if it is affected, what is the clinical signs and symptoms with which the patient is going to present us with. As I already told you, in this question already they have told us neuroimaging studies have shown atrophy of the frontal and temporal lobes. So when there is atrophy of frontal and temporal lobe, it will lead to frontotemporal dementia. In frontotemporal dementia, the executive function is lost. That is planning, organizing, even personality changes, all this will happen. Okay, so that is what we are seeing here in our patient. Suppose visual spatial processing is lost, means how will the patient present to us? They will have difficulty in visual memory and spatial organization, which is mainly the function of the parietal lobe, usually seen in diseases like Alzheimer's disease and stroke. Executive function we already saw is the function of the frontotemporal lobe. Executive functions are lost like planning, judgment, behavior, all these things. What about memory retrieval? Memory retrieval is a function of the hippocampus as well as the temporal lobes. Again, the patient will complain of difficulty in recalling faces, difficulty in recalling events, memory loss will be there, mainly seen in diseases like Alzheimer's and amnesia. Finally, language comprehension is lost, means damage should be either due to the Broca's area, Wernicke's areas, all those things we just now discussed, and they are going to have aphasia. Okay, usually seen in stroke or even in Alzheimer's disease. We'll just take a short break and we'll be back soon. Welcome back. Now let us look at the next question. Which neurotransmitter is primarily involved in regulating mood, sleep, and appetite? Direct question actually. Mood, sleep, and appetite. When you see these three words, it is nothing other than serotonin. Okay. Mood, sleep, appetite directly goes to serotonin. Okay. And see, they've given you a clue also, and is often targeted by antidepressant medication. What antidepressant medication they are talking about? SSRIs, selective serotonin reuptake inhibitor. Okay, so here they have given us 4 neurotransmitters: dopamine, norepinephrine, serotonin, and GABA. Okay, let us look at the function. So here the answer is obviously serotonin. Let us look at the functions of other neurotransmitters which are given here. Okay, dopamine. What does dopamine do? Dopamine is very important for reward, motivation, and motor control. Dopamine is a neurotransmitter for motivation, reward, and motor control. Norepinephrine is for alertness, arousal, and stress response. Alertness, arousal, stress response is for norepinephrine. Okay. GABA. GABA is an inhibitory neurotransmitter. It reduces neuronal excitability. Okay, so that is in brief the primary role of the other three neurotransmitters. We have an entire chapter covered on neurotransmitter, their function, and what are all the disorders that will happen when the neurotransmitter is deficient. Please go through our Medico app. In that, a detailed discussion of all these things is given. So now here for today's discussion, I have briefly summarized the maximum questions which are asked in neurotransmitter topic. One is the functions of these neurotransmitter. Another, types of neurotransmitter. It comes under what category? Even that question has been asked once or twice, I've seen. So first let us see what is a neurotransmitter. Neurotransmitter is a chemical substance that is liberated at the nerve ending, which transfer the message or the nerve impulse from one neuron to the other neuron. That is from the presynaptic neuron to the postsynaptic neuron. That is called as a neurotransmitter. The action of a neurotransmitter is short-lived. We have something called as neurohormone also. Then what is a neurohormone? All these terms are interchangeable. They overlap, but still you should know these terms. Okay, neuromodulator. Okay, a neurohormone is basically a chemical substance secreted by an endocrine gland which is going to act at the synapse and bring, that is, which is going to act on the neuron and bring about similar action. Only thing is, and the effect of the neurohormone takes a longer time. And neuromodulator is nothing but it never directly inhibits or activates, but it is facilitates the activity of a neurotransmitter. So just like similar to co-transmitter, that is a neuromodulator. Now, what is the criteria for a substance to be named as a neurotransmitter? What are the criteria? First, the chemical must be synthesized in the neuron that is concerned. The chemical must be synthesized in the concerned neuron. It must be stored in the presynaptic ending. It must be released at the synapse. A specific receptor should be present for that hormone at the postsynaptic membrane, and it should be disposed by suitable mechanism as soon as it is over. So all these 5 criteria has to be fulfilled by a chemical substance to be named as a neurotransmitter. Okay, then classification. What are questions? Many questions have come from classification as well. How can we classify a neurotransmitter? It can be classified based on its chemical nature, based on its size. So based on its chemical nature, it can be classified as acetylcholine. Acetylcholine per se is a separate entity. Okay, then amine group of neurotransmitter. Under amine group only we have norepinephrine, epinephrine, dopamine, serotonin, histamine. All this comes under amine groups. Then we have amino acid groups. Under amino acid groups we have excitatory and inhibitory amino acid. Excitatory are mainly glutamate and aspartate. Inhibitory are glycine and GABA. Glycine and GABA are inhibitory amino acid neurotransmitter. Glutamate and aspartate are excitatory amino acid neurotransmitter. Then polypeptides, larger in size. Polypeptides, we have antidiuretic hormone, oxytocin, tachykinin. So this ADH, oxytocin and all comes as a neurohormone. Example of a neurohormone we saw just now. Tachykinins like substance P, sorry, corticotrophin releasing hormone, thyrotrophin releasing hormone, GnRH, gonadotrophin releasing hormone, somatostatin, endorphin, enkephalysin, calcitonin gene related peptide, CGRP, neuropeptide Y, angiotensin, activins and inhibins. All these are polypeptide in nature. Then we have purines, adenosine and adenosine triphosphate. Even ATP is a neurotransmitter, and gases like nitric oxide and carbon monoxide. You should be wondering why carbon monoxide is coming here. Even carbon monoxide in under physiological doses is a helpful neurotransmitter in the brain. Okay, it does have physiological functions. That is based on the chemical composition. Now, classification based on the size of the molecule. Based on the size of the molecule, we have large molecule neurotransmitter and small molecule neurotransmitter. Large molecule neurotransmitter, obviously neuropeptides will be coming under it. Small molecule neurotransmitter are all these amino acids, amines, acetylcholine, all this comes under small molecule neurotransmitter. Then co-transmitter. What is a co-transmitter? It is not a neurotransmitter, but it is secreting. It is a chemical that is secreted along with the neurotransmitter, and this is going to facilitate the action of the neurotransmitter at the synapse. Example, release of vasoactive intestinal peptide with acetylcholine. So here VIP is the neuromodulator, which is going to modulate the function of acetylcholine. Similarly, neuropeptide Y along with norepinephrine. So neuropeptide Y is a co-transmitter. Okay. Now coming to serotonin, we already saw. Now going to the next question. We have a 48-year-old patient who presents with symptoms of ataxia, tremor, dysmetria. So the patient has ataxia is there, tremor is there, dysmetria is there. Okay, dysmetria is inability to control the range of movement. So all these three are classical features of lesion where cerebellar, sorry, ataxia, tremor and dysmetria. All this is classical of a cerebellar disorder. And to make our job easier, they have already given here, neuroimaging reveals a significant atrophy of the cerebellum. Which of the following pathways or structures is most likely disrupted here? So we already know there is a cerebellar disorder. Whenever you see this word rate, range, force, direction, okay, the moment you see cerebellum, you have to remember these keywords: rate, range, force, direction, and termination. Rate, range, force, direction, termination. If you see any of these words, you have to think about cerebellum, because the cerebellum has a role. We all know cerebellum is for motor activity. When we are talking about motor activity, it has a role in all these things: rate, range, force, direction and termination of movement. Everywhere cerebellum has a role. Okay, so now let us look at the functional divisions of cerebellum. And then what are the important nuclei through which it brings about its function, and then what happens during cerebellar disorder. Okay. So now coming to the functional divisions of cerebellum, we know cerebellum has three functional divisions: vestibulocerebellum, spinocerebellum, cerebrocerebellum or neocerebellum. Okay, vestibulocerebellum, from the name itself you know, vestibulocerebellum has connection to the vestibular nuclei. That is why it is called as vestibulocerebellum. And the vestibular apparatus is important for maintaining the equilibrium, gait, and learning induced changes in the vestibulo-ocular reflex. So for all these things you need your vestibulocerebellum. And the vestibulocerebellum is made up of the flocculonodular lobe. Each and every line that is mentioned here is a potential MCQ. Okay, so vestibulocerebellum is the oldest, phylogenetically it is the oldest part of the cerebellum. It is made up of folliculonodular lobe. It is concerned with posture and equilibrium. It has connections with the vestibular nuclei, and it outputs directly goes to the brainstem. This is with regards to vestibulocerebellum. Next is the spinocerebellum. Spinocerebellum has two parts. Just like vestibulocerebellum has flocculonodular lobe, spinocerebellum has two parts. One is the vermis, another is the paravermal region. This spinocerebellum receives sensory inputs from all part of the body via the spinal cord to it. That is why it is called a spinocerebellum. What type of sensory input? Proprioceptive inputs. Okay, most of the all the proprioceptive inputs from different parts of the body arrives to the cerebellum, specifically to the spinocerebellum through the spinal cord. Along with this, the motor plan from the motor cortex is also arriving to the spinocerebellum. Now, what it is going to do, it is going to compare both the things. Information regarding the motor plan is also coming. Proprioceptive inputs is also coming. It is the spinocerebellum plans accordingly. So it is very important. Spinocerebellum is very important for smoothness of movements, coordination of movements. So when you see all these words, smoothness of movements, coordination of movements, immediately we have to think about spinocerebellum. Now, if you recall our patient, our patient is having ataxia, our patient is having dysmetria, right? Range of movement is affected. Okay, there is no smoothness in the movement, there is no coordination in the movement. Why? Because the spinocerebellum. So now you know, even without seeing the options, we can say in this patient, the spinocerebellum is affected. Okay, and the output of spinocerebellum comes out through emboliform, fastigial, or globus nuclei to the, from there it goes to the brainstem. These nuclei also you're supposed to remember. Then neocerebellum, also called as cerebrocerebellum. Why is it called neo? Because it is the newest one to develop. Phylogenetically, it is the newest one to be formed. It is also called a cerebrocerebellum because of its extensive connection with the cerebral cortex. This cerebrocerebellum have two cerebellar hemispheres on each side. As I told, it interacts with the motor cortex and it is important for planning and programming movements. Okay, sorry. So neocerebellum is very important for planning and programming of movements. The output from neocerebellum comes through the dentate nucleus and goes to the ventrolateral nucleus of the thalamus. We'll take a break and come back and discuss this question. Okay, welcome back. So what were we doing? We were discussing the different functional divisions of cerebellum. So thus to summarize, we have three functional divisions of cerebellum: vestibulocerebellum, spinocerebellum, and cerebrocerebellum. Vestibulocerebellum has connections with the vestibular nuclei and it is important for balance and eyeball movements. Vestibulocerebellum is made up of flocculonodular lobe. Okay. Then we have the spinocerebellum, which is made up of the vermal and paravermal regions. The vermal region is important for maintenance of posture. The paravermal region is important for skilled voluntary movements. Then we have the neocerebellum on both the sides, which is important for motor planning and action processing. Okay. So as I already told you, when we are discussing about the functions of cerebellum, it has a role in all aspects of movements, starting from the rate, range, force, and direction to until termination. So, postural balance, vestibulocerebellum; smoothing and coordination of movements, spinocerebellum; posture is for the vermal portion of the cerebellum; skilled voluntary movements is the paravermal portion of the spinocerebellum; planning and programming of movements is a function of the neocerebellum. Okay, now how does cerebellum control muscle tone and stretch reflexes? It actually influences the activity of the medial descending pathways. Okay, especially through the vestibulospinal and reticulospinal tract. Okay. By affecting these tracts only, it brings about the muscle tone and stretch reflexes. So if they ask you, how does cerebellum control muscle tone and stretch reflexes? By especially influencing the vestibulospinal via the vestibulospinal and reticulospinal tracts. Okay. Then it is important for learning and improvement of a motor skill. It is important for control of movements on the same side of the body. Since decussation happens twice in case of cerebellum, the movements, loss of movements will be on the ipsilateral side. Okay. Then cerebellum is important to prevent overshoot, and it is important for dampening the movements. That is why in cerebellar lesion you are having intentional tremors. In cerebellar lesion usually there is presence of intentional tremors. In lesions of basal ganglia you have unintentional tremors. So in case of cerebellar lesion, why do we have intentional tremors? The patient tries to do an activity. Suppose they are trying to reach out an object. What are they doing? Because of this loss of dampening movement and overshoot, the patient is going to overshoot. They are not going to reach the target, but they are going to overshoot. Immediately what will happen? Corrective mechanisms will come into place. Now when the corrective mechanisms are activated, now what is going to happen is, again when the patient tries to reach out, now with the corrective mechanism, still the cerebellum is not functioning properly. What is going to happen is, instead of reaching out to the object, now they are going to go in the opposite direction. When this happens repeatedly, the muscle is going to get tired, and which is presented as tremors, intentional tremors. When they are trying to do an activity, tremors are set in. Okay, now you know the reason for intentional tremors in cerebellar disorder. And also cerebellum is very important for the visual judgment of distance. Visual judgment of distance, very, very important function of cerebellum. Okay. Now, how do a cerebellar disorder present? What are all these signs and symptoms a patient is going to do? A variety of signs and symptoms, an array of signs and symptoms are there. I have listed out important features which you should remember. Okay. One, in cerebellar disorder, there is no paralysis. Please remember, in cerebellar disorder, paralysis is not a prominent feature. Voluntary movements are intact, though they are defective. It is intact. That is one point. Second thing, similarly, reflexes are also usually preserved, except it is normal, except for knee jerk, where we can see a pendular knee jerk. Okay. Then similarly, no sensory deficit will also be seen in case of a cerebellar disorder. Hypotonia is a prominent feature in cerebellar disorder, and definitely ataxia will be present. Okay, we have cerebellar ataxia and sensory ataxia. And what is a test that is used to differentiate between sensory and cerebellar ataxia? Romberg's test. Okay, Romberg's test is used to differentiate between sensory and cerebellar ataxia. Okay, so what is ataxia? It is nothing but a motor deficit which occurs in a cerebellar disorder where the coordination of movement is lost. Ataxia is nothing but loss of coordination of movements. Okay, so how is ataxia going to manifest? And also intentional tremors will be present. I already told you. So ataxia in case of cerebellar disorder will manifest as drunken gait and scanning speech. Ataxia in cerebellar disorder is usually manifested as drunken gait and scanning speech. The patient will have a wide, unsteady gait, and scanning speech will be there. Slurring of speech will be present. Okay. We'll take a break and we'll come back with the next question. Welcome back. So what were we discussing? We were discussing about the functional divisions of cerebellum. What is the function of each of the divisions of cerebellum, and how a patient with a cerebellar disorder is going to present. So finally, to finish the discussion, we'll just see what is meaning of dysmetria. Dysmetria is when a patient attempts to touch an object, usually the hand overshoot instead of reaching the target. This is called as dysmetria. Okay, so now you know what is the answer for our subject. He has difficulty in coordination of the movement, inability to control the range of motion. So this is the function of what? The spinocerebellar tract. Okay, then what about corticospinal tract, basal ganglia, and medial lemniscus? The corticospinal tract is important for transmitting the motor commands from the motor cortex to the through the spinal cord to the peripheral structures. So now when the corticospinal tract is affected, typically you will see paralysis and muscle weakness. Usually the related condition would be patient of multiple, they will say a patient of multiple sclerosis or a patient of stroke presented with muscle weakness. What do you think is the structure the or pathway that is affected? Then you will select corticospinal tract. If they are saying a patient is having bradykinesia, it is having unintentional tremors, or tremors at rest, okay, has difficulty to regulate his motor control and coordination of movement is lost, then what is the structure that is involved? Basal ganglia. Medial lemniscus. Medial lemniscus, what is the information? It conveys sensory information like touch, proprioception to the thalamus and then to the higher center. So if medial lemniscus is affected, what is the thing? Sensory system is lost. Proprioception is lost. Loss of touch, loss of proprioception. Such information will be given by the subject. And this also happens in conditions like stroke and encephalopathy. Okay. In basal ganglia lesion, diseases like Parkinson's disease, Huntington's disease will be described. Okay, is it clear? If the corticospinal tract is affected, most of the time the disease they mention would be either stroke or multiple sclerosis, and the symptom of the patient is going to be muscle weakness or muscle paralysis. If basal ganglia are affected, the disease they will mention is either Parkinson's disease or Huntington's disease, and the symptom the patient is going to have are unintentional tremors, bradykinesia, slowing of movements, all those things. Medial lemniscus means it is totally sensory system. Fine touch, proprioception will be lost. Okay, so that is with that question. We will move on to the next question. We have a 30-year-old patient. He presents with episodes of transient loss of consciousness and involuntary movements. So we have a 30-year-old patient presenting with episodes of transient loss of consciousness and involuntary movements. Electroencephalogram is performed to evaluate the underlying cause. Loss of consciousness, involuntary movements. Okay. EEG is showing spikes and sharp waves were predominantly in the temporal lobe. Based on this finding, which of the following condition is most likely associated with the EEG abnormality? So to answer this question, we should know briefly you have to learn about the different types of epilepsy. Okay, and what are all the EEG finding, cardinal or important single best EEG finding in each of those different types of epilepsies. Only when we know that we will be able to answer such questions. In this question, even if you don't know, there is a clue, you can easily answer this question. What is the clue? They have told you the temporal lobe is affected. Okay, from the temporal lobe only, the spikes and sharp waves are recorded. So obviously the answer here will be temporal lobe epilepsy. Okay, but all questions are not going to be this easy. It is better for us to read and understand what are all the EEG findings in classical EEG findings in all these 4 different types of seizures. Okay. So in our question, we had spikes and sharp waves seen recorded from the temporal lobe. So very classical of temporal lobe epilepsy. How will the description of that EEG be? Usually the report will be mentioned as focal epileptiform discharges seen from temporal lobe, which is having spikes and sharp waves. If you see a description like that, we are sure it is temporal lobe epilepsy. Suppose we have a description of diffuse high voltage synchronized activity, like that of a spike and wave pattern. It is indicative of generalized tonic-clonic seizures. Okay. Then we have a description where they are saying briefed episodes of impaired consciousness along with a 3 hertz spike and wave discharge. This is very classical. 3 hertz spike and wave discharge means it is absent seizures. And irregular spikes with brief sharp like shock like muscle jerks. That means they are talking about myoclonic seizures. Okay. I hope this once you memorize this table, you will be able to answer questions coming from EEG. Okay, now we have another question. A 60-year-old patient with chronic obstructive pulmonary disease presents with elevated arterial blood carbon dioxide level. We have a COPD patient. Okay, this COPD patient is having hypercapnia as well as hypoxia, increased blood carbon dioxide level and decreased blood oxygen levels. Which of the following physiological mechanism is most directly impaired in this patient, leading to the observed gas exchange abnormality? So we have a patient of COPD presenting with hypoxia and hypercapnia. Okay, because of this hypoxia and hypercapnia, what is going to happen? That is what they are asking. What physiological process has been impaired? Is it the oxygen hemoglobin dissociation curve, or is this the alveolar ventilation, or is this the carbonic anhydrase activity, or is it the hemoglobin binding to oxygen that is impaired? The obvious answer here is alveolar hypoventilation. Why am I telling it as alveolar hyperventilation? Because we have two important features in the patient: hypercapnia, hypoxia. When you look at these two features, what information does it give? Hypercapnia and hypoxia both are present. That means gas exchange is not happening. Adequate gas exchange is not happening. Why adequate gas exchange is not happening? Because COPD is a chronic disease where the airways, obstruction of airways is there. So air entry is limited. Because the air entry is limited, alveolar ventilation. What is alveolar ventilation? Exchange of gases. So the alveolar ventilation will get affected. When alveolar ventilation is affected, obviously carbon dioxide is not leaving the lungs, oxygen is not entering into the RBC, right? Leading to both hypoxia as well as hypercapnia. That is why the best option for this is alveolar ventilation is affected. Okay. What about the other options? They have given oxygen hemoglobin dissociation. In case of COPD, the oxygen hemoglobin dissociation curve will be shifting towards the right. Okay. And also the related conditions would be different. They would have either given as anemia or a respiratory alkalosis, like that. What about carbonic anhydrase activity? What is carbonic anhydrase? It is an enzyme which catalyzes the formation of carbonic acid from carbon dioxide and water. So here, when we are talking about carbonic anhydrase deficiency, the clinical scenario will be something like that of a acid-base disorder. Okay, not something directly related to COPD. And finally, binding of oxygen to hemoglobin. When this capacity is impaired, then also the clinical scenario what they give would be more towards a sickle cell anemia, like that, rather than COPD. Okay, is it clear? Or some abnormal forms of hemoglobin, those type of conditions will be described rather than COPD. Okay, I hope now it is very clear why in this patient of COPD it is the alveolar ventilation that is affected rather than the other pathologies that they have made, other physiological processes that they have mentioned here. Okay, now we have our next question. A 52-year-old patient with COPD again. So earlier patient was also a COPD patient. This is also a COPD patient. Probably we tend to have a lot of COPD patients, right? So now we have a 52-year-old male with COPD presenting with worsening of shortness of breath. So dyspnea is getting worsened. An ABG reveals a reduced partial pressure of oxygen. Okay, despite oxygen therapy. Then what else is there? So we have a COPD patient. He is worsening. He has worsening dyspnea. So the patient is put on oxygen. Even after giving oxygen, still his partial pressure of oxygen has not improved. Okay, so this much we know till until here. The oxygen hemoglobin dissociation curve is likely to be affected in this patient. Definitely, I told you just now, no? In case of COPD patient, the oxygen hemoglobin dissociation curve will be shifted towards the right. How does oxygen hemoglobin dissociation curve shift in response to chronic hypoxia as seen in COPD? What is the answer? I've been telling the answer from the previous question itself. What is the answer here? The oxygen hemoglobin dissociation curve will be shifted towards the right, decreasing the oxygen affinity. So that is the answer here. Okay, but why is this happening? How is this happening? That you should know. Okay, so what happens here? Again, in COPD, as we saw just now, alveolar ventilation is affected. Since alveolar ventilation is affected, there is both hypoxemia as well as hypercapnia. A chronic condition like hypoxemia is going to increase the production of 2,3-DPG in the RBC. We all know RBC lacks mitochondria. So, what type of cellular respiration will happen in case of RBC? RBC is not using oxygen, so it is having anaerobic glycolysis, right? So now, in such a scenario of chronic hypoxia, the RBC and the production of 2,3-DPG in RBC is going to increase. And we all know increase in 2,3-DPG is going to shift the curve towards the right. That is one of the reasons why in chronic COPD or in patients with chronic hypoxia, the oxygen hemoglobin dissociation curve will shift towards the right. But that is not the only reason. If you look at COPD, what are the other reasons why the curve is shifting towards the right? We should know. One is hypoxia. Okay. Second thing, in a condition, chronic condition like COPD, the metabolic demands is more. The patient is dyspneic, he's expending a lot of energy. The metabolic demands are more. So, whenever the metabolic demands are more in a structure, then temperature will increase. So, there is increase in temperature. Increase in temperature will also shift the oxygen hemoglobin dissociation curve towards the right. And finally, hypercapnia is there because of the impaired alveolar ventilation. There is hypercapnia. Hypercapnia means what? Increased partial pressure of carbon dioxide. This carbon dioxide is going to cause increase in partial pressure of carbon dioxide is going to cause respiratory acidosis. So respiratory acidosis means what is going to happen to the pH? The pH is going to drop. Okay, so there is acidosis, there is increase in temperature, there is increase in 2,3-DPG. Decrease in pH, increase in temperature, increase in 2,3-DPG. All these are factors that is going to shift the oxygen hemoglobin dissociation curve towards the right. Okay, now I hope you are clear why the oxygen hemoglobin dissociation curve is shifted towards the right in case of patients with COPD. Okay, now is this shifting good or bad? This shifting is beneficial to the patient. That is why you see in that option, it gets shifted towards the right, decreasing the oxygen affinity. Yes, why? Because once the affinity of oxygen to hemoglobin is decreased, then only what will happen? Unloading of oxygen. This is the key word. Please remember. Unloading of oxygen will happen. So in hypoxia, the right shift is called as, this is called as Bohr's effect. The right shift of the oxygen hemoglobin dissociation curve is called as Bohr's effect. Okay, in Bohr's effect, what is happening? The right shift leads to decreased affinity of hemoglobin for oxygen, so that more oxygen is available for the tissues. Okay, clear? This is a helpful. That is why in this patient, despite giving oxygen therapy, because of the rightward shift, there is unloading of oxygen or decreased affinity of oxygen, decreased affinity of hemoglobin for oxygen. That is why despite giving O2 therapy, the partial pressure of oxygen of this patient is low. Okay, that means the partial pressure of oxygen is low, the tissues are able to take up the oxygen. Okay, I hope that clears most of your discussion on COPD. Okay. So here are the entire list of factors that is shifting the curve towards the right and left. Important ones, what you have to remember in case of an oxyhemoglobin dissociation curve is, anything that shifts the curve towards the right is going to cause unloading of O2. Right side means unloading. Left side means loading of O2. Okay, that is there. Most important, what are the things? pH, acidosis, increase in temperature, increase in 2,3-DPG level, hormones like growth hormone, androgen, thyroid hormone, high altitude, hypoxia, exercise, anemia. All this will cause a rightward shift. Just the opposite, fetal hemoglobin, alkalosis, stored blood, decrease in 2,3-DPG, decrease in PCO2, decrease in temperature is going to shift the curve towards the left. Okay, so those are the factors that is shifting the curve. Now let us discuss little bit about this oxygen hemoglobin dissociation curve itself. Okay, so what you see here is the oxygen hemoglobin dissociation curve. Okay, I hope it is visible. So we have the oxygen hemoglobin dissociation curve here. In the x-axis, what is present in the x-axis? In the x-axis, you have the partial pressure of oxygen. In the y-axis, what do you have? Percentage saturation of hemoglobin with that of oxygen. So what is this oxygen hemoglobin dissociation curve per se? Basically, this is a graphical representation of the partial pressure of oxygen and the percentage saturation of hemoglobin at that particular partial pressure of O2. Okay, now what you see is, this is the normal curve. This is the curve that is shifted towards the right. This is the curve that is shifted towards the left. Left, right, normal. Okay, so we have the right shift curve, the left shift curve, and the normal curve. First, let us look at the normal curve. Okay, what is the shape of this normal curve? Is it like this or is it like this? Okay, we have the S-shaped curve. So we have a S-shaped curve, which is also called as a sigmoid shaped curve. Very important. First thing they will ask us, what is the shape of this curve? The oxygen hemoglobin dissociation curve is not linear, but it is sigmoid in shape. Okay, it has two phases. One is the steep phase. Then you have the plateau phase. It has a steep phase and a plateau phase. First, why is this curve not linear and why is it sigmoid shaped? This curve is sigmoid in shape because of the manner in which, okay, we call it as cooperative binding. Because of the cooperative binding of oxygen with hemoglobin, or the cooperative binding of the affinity of oxygen for hemoglobin. Why? What do we mean by cooperative binding? Cooperative binding means when the first molecule of oxygen binds with hemoglobin, it increases the affinity of the second molecule. When the second molecule is also bound, then the affinity of the third molecule is much more increased than the second. So this is like a positive feedback mechanism until all four molecules are bound with oxygen. That is when the hemoglobin is completely saturated with oxygen. So this type of binding is called as cooperative binding, and because of the manner, this type of cooperative binding, the shape of the curve is not linear, but it is sigmoid. It has a steep face and then plateau face. Okay, this much is it clear so far? Okay, now let us look at the other features of this curve. Okay, so what is the reason for this steep face? When we say steep face, it is because of the cooperative binding nature of oxygen to hemoglobin. Because of that, then we have a plateau phase. So then you have these dotted lines. What are these dotted lines? These dotted lines are nothing but it is showing the P50. Okay, P50 is nothing but the partial pressure of oxygen at which 50% of hemoglobin is saturated with oxygen. Okay, so in case of a normal curve, the P50 is around 27 millimeters of mercury. The normal value of P50 in case of a normal curve is, okay, that means at a partial pressure of 27 mmHg, 50% of the hemoglobin is saturated with oxygen. So this is the 100%. Okay, this is not 100. So this is 100%. So this is a place where it is 50% saturated. So that is called as P50. When there is a rightward shift, the P50 increases. The P50 value increases for a rightward shift. In case of leftward shift, the P50 decreases. Okay, so whenever you see the line P50 decreases, that means you have to immediately understand that there is a leftward shift. Whenever they are mentioning the P50 increases, that means there is a rightward shift. Rightward shift means unloading of oxygen to the tissues. Leftward shift means loading of oxygen to the hemoglobin. Okay, this much is clear. Okay, now let us look at the reason why this one phase is steep and the other phase is flat. If you see, the plateau phase usually starts, the plateau phase usually starts when the partial pressure of oxygen has crossed 60 mmHg. Once the partial pressure of oxygen has crossed beyond 60 mmHg, the curve flattens out. Okay, from 60 mmHg to around 100 mmHg, the oxygen saturation, the percentage saturation of hemoglobin is only around 10. That is between 90 to 100%. By 60 mmHg, for a partial pressure of oxygen of 60 mmHg, almost already 90% of the hemoglobin is saturated with oxygen. From 60 to 100, only the remaining 10% of saturation only happens. What is the significance of this plateau phase? So that tells us that even in a wide range of fluctuations in partial pressure of oxygen, that is from 100 to 60, a drop of partial pressure of normal partial pressure of oxygen is anything above 95 mmHg. We usually keep and see, right? Pulse oxy, anything above 95 is normal. But from this 95, even if it drops to partial pressure of oxygen drops to 60 mmHg, still the percentage saturation is only going to drop around 10%, that much only. So still the patient is not going to have hypoxia. Now, the 60 mmHg, beyond 60 mmHg, what you see is the steep phase, steep rise or steep fall. We can say any of this. So the 60 mmHg is a critical value. 60 mmHg is the critical value for the partial pressure of oxygen. Once the partial pressure of oxygen drops below 60 mmHg, then we will come into the steep phase. Okay, so what will happen? Even after 60 mmHg, even a slight drop in the partial pressure of oxygen, suppose from 60 to it comes to 50 mmHg. If you look at the curve, because if it is steep, look at the drop. The curve is very steep. So for a below 60 mmHg, even a little bit of change in the partial pressure of oxygen is going to cause greater unloading of oxygen from the hemoglobin. Greater unloading of oxygen from the hemoglobin will happen. For change from 60 to 100, hardly 10 percentage will only change in the saturation. But after that, see, if another 10 mmHg drops below 60, the percentage is more than 30%. Okay, here. Why? Because 60 mmHg is a critical value. When it drops below the critical value, even a slight drop will cause larger unloading of oxygen. That is the significance of the steep phase. Okay, now I hope it is clear for you. With this, we will move on to the next question. Okay. Okay. So what is this question? Which of the following condition is characterized by central cyanosis due to decreased oxygen content in the arterial blood? Okay. What they are asking is, what is the cause of central, which among these four conditions will cause central cyanosis? Okay. So first we have to remember, first we have to read what is cyanosis. Cyanosis is nothing but bluish discoloration of the skin and mucous membrane. Cyanosis is nothing but bluish discoloration of the skin and mucous membrane. Why should it become blue in color? Whenever the amount of deoxyhemoglobin whenever the amount of deoxygenated hemoglobin in the blood increases, it gives a bluish tinge. That is why oxygenated hemoglobin will give a pink tint to the skin. When deoxyhemoglobin level increases, it gives a bluish tint to the skin. That is why it is called as cyanosis, is called as bluish discoloration of skin and mucous membrane. When can cyanosis happen? When the amount of deoxygenated hemoglobin or reduced hemoglobin exceeds 5 gram percent. When the deoxyhemoglobin level exceeds 5 gram percent, we call it as cyanosis. Okay, and at the same time, the arterial blood oxygen saturation will be below 85%. So for this patient to have central cyanosis, the oxygen content is decreased. That means definitely it is going to be less than 85 percentage. Okay, the arterial oxygen content, and also cyanosis is there. So the deoxyhemoglobin is going to be greater than 5 gram percent. Then only this patient will have cyanosis. Okay. There are two types of cyanosis. Okay, one is the central cyanosis, the other is the peripheral cyanosis. Here already they have mentioned the patient is having central cyanosis. So let us see what is central cyanosis. What are all the diseases where we can see central cyanosis? Central cyanosis is a systemic issue affecting the oxygenation of blood, typically presenting with bluish discoloration in the lips, tongue and core of the body. So in central cyanosis, where are we going to find the bluish discoloration? In the lips, tongue and core of the body. When the cyanosis is very severe, then peripheral parts will also be affected. Causes for central cyanosis. Respiratory causes. What are all the respiratory causes for central cyanosis? COPD, asthma, pneumonia, pulmonary embolism, interstitial lung disease. In all these causes, there will be central cyanosis. I'll repeat, central cyanosis is seen in COPD, in asthma, in pneumonia, pulmonary embolism and ILD, interstitial lung disease. Now, what are all the cardiac causes? Congenital heart disease, tetralogy of Fallot, left to right shunt, that is patent foramen ovale, heart failure. Basically, whenever there is shunting of blood, admixture of arterial and venous blood, it will result in central cyanosis, definitely. So, in cardiac conditions, it is going to result in central cyanosis. Hypoventilation due to a neuromuscular disorder like myasthenia gravis or a drug overdose again will result in central cyanosis. Hemoglobin abnormalities like methemoglobinemia, carbon monoxide poisoning, there also there will be central cyanosis, but in all these cases, in carbon monoxide poisoning, even before the cyanosis, the patient will be very critical. Okay, then comes peripheral cyanosis. What is peripheral cyanosis? Peripheral cyanosis typically will affect the peripheral parts of the body. The extremities will be affected. Why? Because there is either due to a reduced blood flow or there is vasoconstriction of the blood vessels in the periphery. Okay, or increased extraction, decreased blood flow or increased extraction of the blood, oxygen from the blood. This is all going to cause peripheral cyanosis. What are the causes? Circulatory causes like peripheral vascular disease, Reynolds phenomenon, occlusion of the artery, exposure to cold. Patients of heart failure who are having poor perfusion, patients of septic shock, cardiogenic shock, and conditions like intense exercise where there is more extraction of oxygen from the blood. All these will result in peripheral cyanosis. So, if I have to summarize, central cyanosis is a problem with the systemic oxygenation of the blood. So that mainly the core parts of the body like nose, tongues, lips, earlobes will be affected. Central cyanosis will be present there. Whereas peripheral cyanosis is because of a local circulatory issue, so the extremities will be blue in color. Okay, so now it will be easier to answer this question. Our patient is having central cyanosis. What is the which among these four diseases will cause central cyanosis? Is it COPD? Or is it congenital heart disease? Is it peripheral vascular disease? Or is it Reynolds phenomenon? Okay, actually here congenital heart disease with right to left shunt is the most apt answer. Next to this only COPD will be there. So in this question the best answer will be, see, these two, peripheral vascular disease and Reynolds phenomenon, will cause peripheral cyanosis. Okay, already I told you respiratory cause of central cyanosis is COPD, but when we come uh, in the order of importance, congenital heart disease with shunting will be the first best option, followed by COPD. Okay, the next question. We have a preterm neonate who is diagnosed with respiratory distress syndrome. This condition is primarily due to immaturity of the lung and insufficient surfactant production. So they have told the disease and they have told the cause also. The child is having, infant is having, neonate is having RDS because of insufficient surfactant production. Which of the following is the primary role of surfactant in the alveoli? What is surfactant? Surfactant is a surface active agent which is going to reduce the surface tension. We know this. This is a very direct question. So what are the options they have given you here? For A, it regulates the blood flow to the lungs. B, it increases the surface tension within the alveoli. C, it reduces the surface tension within the alveoli. D, it enhances alveolar ventilation. What is the right answer? Surfactant reduces the surface tension within the alveoli, thus preventing the collapse of the alveoli. Correct? Right. Now let us discuss about surfactant. So already I told you surfactant is a surface active agent which acts by decreasing the surface tension, thus preventing the collapse of the lung. It is, what is the source of surfactant? Surfactant is produced by the type 2 alveolar epithelial cells, also known as type 2 pneumocytes. So what you are seeing here in this picture is a type 2 alveolar pneumocyte. Even in this type 2 alveolar pneumocyte, you are seeing something called as lamellar bodies. What is this LB? Lamellar bodies. These are inclusion bodies. These lamellar bodies contain surfactant. So this is an important MCQ. Surfactant is present in the lamellar bodies of your type 2 alveolar pneumocytes. How is it synthesized? For production of surfactant, you need certain substrate molecules like palmitate, choline, glucose, etc. All these substrate molecules are picked up by the type 2 alveolar pneumocyte. After that, what it is doing? It is going to synthesize dipalmitol phosphatidylcholine, DPPC, dipalmitol phosphatidylcholine, from these substrates where it is happening in mitochondria. So DPPC is produced in mitochondria, synthesized in mitochondria. This surfactant not only contains dipalmitol phosphatidylcholine, but it also contains proteins. Okay, so from where are these proteins produced? Normally, protein is synthesized from the endoplasmic reticulum, right? So now mitochondria is synthesizing DPPC, endoplasmic reticulum is synthesizing protein. Now these are getting, both of this is getting packed in the Golgi apparatus. Then these materials are stored in the lamellar bodies. Whenever it is, now from the lamellar bodies, by the process of constitutive exocytosis, it is released into the alveolar epithelial surface. Okay. What do you mean by constitutive exocytosis? Okay, constitutive exocytosis means unregulated exocytosis. The process is going to happen continuously. Hormones are all non-constitutive or regulated exocytosis. That is, whenever it is needed, it will be released. Whereas surfactant, as it is continuously produced and it is released. So such a process is called as constitutive exocytosis. Once it is released, it is going to form a phospholipid film on the alveolar epithelial surface, reducing the surface tension. So that is the steps involved in synthesis of surfactant. Composition already we saw. It is mainly made up of dipalmitol phosphatidylcholine along with proteins. So 90% of the surfactant is made up of lipids. Even their most important lipid is DPPC, followed by phosphatidylglycerol, phospholipids and neutral lipids. 8% is protein. What are the proteins present in surfactant? Albumin, immunoglobulin, mainly IgA, and apoproteins like SPA, B, C, and D. What is the function of these proteins? They facilitates the formation of this phospholipid film. And among these upper proteins, SPA controls the reuptake of surfactant by type 2 cells. Reuptake, reuse, it helps in all this process. And 2% of the surfactant is made up of carbohydrates. What is the time of surfactant synthesis in fetal life? It starts around 28 weeks, that is the 7th month of pregnancy, and it is 90% completed at the time of birth, term neonate. What are all the factors that facilitate the synthesis of surfactant? Glucocorticoid hormones, thyroid hormone, insulin, stretching of the lungs, beta agonist, calcium, and regular physical exercise will facilitate the production of surfactant. What are all the factors that inhibit the formation of surfactant? Premature birth, hypoxia, acidosis, inflammation, mechanical ventilation, pulmonary edema, oxidative stress, malnutrition, smoking, drugs and toxin, all these will inhibit the production of surfactant. Okay, what are the functions of surfactant? Already we know one. What is it? Decrease the surface tension. By decreasing the surface tension, what it is doing? It is preventing the alveolar collapse. It is improving the alveolar stability, prevents pulmonary edema, decreases the work of breathing. All this happens because of the reducing the surface tension, and also surfactant promotes phagocytosis. Okay. Now going to the next question. Which of the following statement about decompression sickness? Which of the following statement about decompression sickness are true? Okay. So what is the first statement? So in decompression sickness, what you have to remember is in deep sea diving, at the level of sea level, it is normal 1 atmospheric pressure. So at the once with when you go every 33 meters down or 10 feet, 33 meters down or 10 feet below the sea level, descent for every 33 meters or 10 feet descent, what is going to happen is the atmospheric pressure is increased by 1 atmosphere. So as we keep diving down, the atmospheric pressure increases. There is high pressure. So what will happen in decompression sickness? Because of the high pressure, as the person goes into the deep sea diving, what are they going to do? They are going to take a mixture of oxygen and nitrogen. So these gases under pressure, since nitrogen is lipid soluble, it will go and dissolve into the various cell membranes, especially the neurons. Then what is going to happen? Decompression sickness occurs due to formation of nitrogen bubbles in the bloodstream and tissues when a diver ascends too quickly from the deep water. Yes, now they have gone deep into the water. All these are dissolved and all the gases are dissolved in the tissues and blood. Now when they are coming up, when the diver is ascending up and when he is ascending too quickly, definitely what is going to happen? The gases that was dissolved is going to bubble out. Okay, if they are ascending too quickly without any safety stops, then is this correct? Yes, this is a correct answer. What have they asked? They have asked us to identify the true statement. Option A is actually true. Then what is the option B? The primary symptom of decompression sickness include joint pain, dizziness, skin rashes. but it does not affect the central nervous system. See, the first half of the symptom is correct, but the statement that it does not affect central nervous system is false. It does affect central nervous system. Symptoms like alcohol intoxicity will be present in case of decompression sickness pertaining to central nervous system. The treatment for decompression sickness involves hyperbaric oxygen therapy, which helps to reduce the size of the nitrogen bubble and facilitate their absorption. Yes, this is also true. Prevention of decompression sickness include adhering to proper ascent rates and conducting safety stops during the ascent from the dives. Yes, this is also true. Okay, so actually this question has to be false. Which of the thing is false? If they've asked like that, then we would have selected option B. But since now they have asked which is true, the best option to select here would be decompression sickness occur due to formation of nitrogen bubbles in the bloodstream, and when the diver ascends too quickly, these nitrogen bubbles keep rushing out. So that is the right answer here. Okay, actually this question itself is wrong. Coming to the next question. Which component of heart's electrical conducting system is responsible for initiating heartbeat and setting the pace for heart rate? So this is again a direct question. They are asking which is the pacemaker of the heart. Which among these four is the pacemaker of the heart? Obviously the SA node. Okay. Why do we say SA node is the pacemaker of the heart? Why not the other? Because the rate at which SA node produces impulse, the rate of impulse generation in SA node is highest compared to the other structures mentioned here. Okay. So now let us look at the conducting system of the heart. We'll just take a break and we'll be coming back. Okay, welcome back. So what we were discussing? We were discussing the conducting system of the heart, and we were seeing which is the pacemaker of the heart. So the question was, which is the pacemaker of the heart? So the SA node is the pacemaker of the heart. Now, why is SA node the pacemaker of the heart? Because the rate of impulse generation is the highest for SA node. So where is this SA node located? It is located in the right atrium near the opening of the superior vena cava. So how is the conducting pathway for the heart? From SA node, impulses are going to go to the AV node after passing through the internodal pathways. Okay, so SA node generates the impulse, and from there it passes to the entire atrium through the internodal pathways, anterior, middle and posterior internodal pathways, and then it reaches the AV node. At the AV node, something important is happening. The AV node delays or pauses the impulse transmission by 0.1 second. AV nodal delay. This is also an important MCQ. AV nodal. How much is the AV nodal delay? 0.1 seconds. Okay, 0.1 second. Why AV nodal delay should happen? Two reasons. One is so that the atrium completely empties the blood and the ventricle completely fills with the blood. Okay, so that is why you need AV nodal delay. Otherwise, the ventricular otherwise the ventricular rate and the ventricular rhythm will be completely it is going to be completely haphazard. So to prevent this and to have a organized systole, contraction and relaxation, we have this AV nodal delay. Okay. Then from the AV node, the impulses are going to go into the bundle of his, then right and left bundle branches, and from there into the via the Purkinje fibers, it is going to go and depolarize the ventricles. So this is the pathway. Now let us look at the rate of impulse generation. Okay, the SA node is the pacemaker of the heart. It sets the rhythm for heart rate. Normally the rate of impulse generation is 60 to 100 beats per minute in case of SA node. Following SA node, AV node. What does AV node do? Already I told it relays impulses from the atria to the ventricles. Two things it is doing. One is AV nodal delay, which is 0.1 second, and second thing, the rate of impulse generation in case of AV node is 60 to 40 beats per minute. Okay, then what about bundle of His and Purkinje fibers? Bundle of His conducts it from the AV node to the ventricles, and Purkinje fibers is going to distribute it towards the entire ventricular myocardium. The rate of impulse generation for bundle of His is around 20 to 40 beats per minute, and Purkinje fiber is also 20 to 40 beats per minute as well. So suppose if the SA node fails, then what will take over? The AV node will take over. Even if the AV node fails, then the bundle of His. Even that fails, the ventricular muscle. Okay. Suppose if the ventricular muscle has taken over, then what will be the heart rate of the patient? Only around 20 to 40 beats per minute. So now you understand why SA node is a pacemaker of the heart, because it has certain cells called as pacemaker cells, which has the function of autorhythmicity. It is going to generate impulse. The other cells are AV node, bundle of His, Purkinje fibers, everything in the conducting system is also capable of sorry, is also capable of generating impulses, but the highest rate of impulse generation is for SA node. That is why SA node is a pacemaker of the heart. So this is one question. Another question which is commonly asked and people confuse is the conduction velocity. Okay, pertaining to conductive system of the heart. One is the rate of impulse generation. The other is the rate of conduction velocity of the different tissues. So that if you see, the highest rate of conduction is there for Purkinje fibers, 4 meters per second. So the impulse is generated. Which among this tissue in this conducting system has the highest rate of impulse conduction? Purkinje fiber. Okay, don't confuse. Conduction velocity is highest for the Purkinje fibers and it is lowest for the SA node. Okay, for SA node it is around 0.05 meters per second, whereas for Purkinje system it is around 4 meters per second. Okay. Okay, now we have another question. A 65-year-old patient with history of hypertension, okay, history of hypertension with the blood pressure reading of 190 by 100, 190 systolic, 110 diastolic, reports to us. Okay, so we have a 65-year-old patient with history of hypertension. He presents with the current blood pressure reading of about 190 by 110 despite being on antihypertensive medication. That is, this patient is being treated for high blood pressure. Still, his blood pressure is not getting control. It is at a range of 190 by 110 mmHg. Now, on examining the patient, he shows signs of left ventricular hypertrophy in ECG. LVH is present in the ECG. Which of the following mechanism is most likely to contribute to the development of left ventricular hypertrophy in this patient? Okay, so here, just like our COPD case, there are certain basic things which I want you to remember. Once these concepts are clear, you are never going to make a mistake in this topic. First thing what you have to understand, even before looking at the options, we need to understand why in a patient of hypertension, left ventricular hypertrophy is happening. Okay, so the answer for that is hypertensive patients will have increased systemic vascular resistance. This we all know. Hypertensive subjects will always have increased, especially for this patient, uncontrolled hypertension. Even on medications, his hypertension is not getting controlled. So this patient is definitely going to have increased systemic vascular resistance, SVR. SVR is increased in this patient. Now, why SVR is increased in an hypertensive? Because questions can come like that also. And they'll give you the reasons, causes for increased systemic vascular resistance in hypertensive individuals is due to, or identify or exclude any one like that. So now, why does hypertensive subjects have increased systemic vascular resistance? Okay, because one is, in hypertensive patient there is vascular remodeling that is happening. What do I mean by vascular remodeling? Vascular remodeling means the smooth muscles of the blood vessel gets remodeled, it gets hypertrophied. Why? Because of the increased pressure in which the blood is flowing through these arteries, they get remodeled and it gets hypertrophied. Once the muscle gets hypertrophied, the vascular smooth muscle, what is going to happen? The lumen, the diameter of the lumen is going to get narrowed. That is one reason. So when the lumen diameter is narrow, there is resistance is going to increase. That is one reason. Now, second reason, in patients of hypertension, there is definitely endothelial dysfunction. Whenever there is endothelial dysfunction, the main contributor for this endothelial dysfunction is lack of nitric oxide. We already saw during the functions of nitric oxide, it is a very, very potent vasodilator, right? So now nitric oxide is not available. So there will be vasoconstriction. Again, the diameter of the blood vessel is low, which increases the systemic vascular resistance. Third, there will be altered autoregulation of blood flow. That is a myogenic theory. We all know when the blood vessel is getting stretched, it is going to contract in response to the stretch. Now, in this chronic hypertension, because of the increased pressure at which the blood is flowing, the myogenic response become abnormal. So the blood vessel is going to constrict a lot. Again, resist because of the narrowing of all this is collectively reducing the diameter of the blood vessel. When blood vessel diameter reduces, resistance is going to increase. And finally, these patients will have increased patients of hypertensives will have increased sympathetic outflow. When sympathetic outflow increases, again there is going to be vasoconstriction. So all these four factors are only mainly and finally increased viscosity of blood as well. But these four factors are very, very important contributing factors for increase in systemic vascular resistance in hypertensives. So now the systemic vascular resistance has increased. Now, what is happening to the heart? The heart has to pump blood against this increased vascular resistance. So, basically, the afterload to the heart has increased. We are increasing the afterload on the left ventricle. The left ventricle, the aorta has to pump against the resistance. So, the afterload to the left ventricle has increased. So, when the left ventricle is pumping against higher than normal load, obviously to maintain the cardiac output, what is going to happen? It is working harder. When it is working harder, it is going to get hypertrophy. So this is the reason for hypertrophy that happens in left ventricular hypertrophy in hypertensive patients. Okay. Other options given here like plasma renin, decrease in blood volume do contribute, but it is the increase in systemic vascular resistance, option A, which is the most direct factor contributing to LVH in this patient. Okay, clear all of you? Now, we have another patient, a 45-year-old patient with chronic kidney disease, experiencing issues with urine concentration. Urine concentration is the function of what? Countercurrent mechanism, right? Okay, so we have a 45-year-old patient who with chronic kidney disease experiencing issues with urine concentration and is having impaired countercurrent multiplication. Okay, see most of the clues already given in the question itself. They are very clearly told it's a countercurrent multiplication which is affected here. Which segment of the nephron is primarily responsible for establishing the osmotic gradient essential for countercurrent multiplication? Okay, and what is its role? That is what they are asking. Okay, either it is a PCT or distal limb of loop of Henle, descending limb of loop of Henle, or the ascending limb of loop of Henle, or is it the distal convoluted tubule? This is also a Direct answer, very easy. Before answering the question, let us discuss briefly about countercurrent mechanism. Okay, what is this countercurrent mechanism? Countercurrent mechanism is one in which you will have two tubes which are parallel to each other, which are in close approximation to each other, and the flow will be in opposite direction. Okay, such type of countercurrent mechanism happens at few places in the human body. Where does it happen? One is in the renal tubules. Then where else? The pampliform plexus of veins in the testis to maintain the temperature of the scrotal tissue. Then where else? In the gut epithelium also. So these are the common places where we can find. These are the places where countercurrent mechanism happens in the human body. Now, as I told you, for countercurrent mechanism, all those three conditions which I told you needs to be fulfilled. We should have two parallel tubes close to each other, and the inflow and outflow should be in opposite direction. Okay, now coming to the kidneys. In kidneys, where do you see such a structure? In kidneys, where do we see such a structure? We are seeing it here, right? That is a loop of Henle. You have the descending limb, you have the ascending limb. They are close to each other. In descending limb, the flow of the fluid is towards the medullary interstitium. In ascending limb, it's away from the medullary interstitium. So our loop of Henle is perfectly satisfying all those conditions. Okay. The countercurrent mechanism which is available in kidney has three components. Okay, 1, 2, and 3. The countercurrent mechanism in kidney has three components: countercurrent multiplier, countercurrent exchanger, and then an equilibrating device. Three things are there. Countercurrent multiplier function. Our question is there specifically for countercurrent multiplier. The function of countercurrent multiplier is done by the loop of Henle. Okay, countercurrent exchange is function of the vasa recta, and the equilibrating device is the collecting duct. Collecting duct acts as a osmotic equilibrating device. Many of the times you will be able to answer a loop of Henle and vasa recta, but when they ask osmotic equilibrating device, you will struggle. So the osmotic equilibrating device is nothing but the collecting duct. All these three are components of countercurrent mechanism. Okay. So now in our question, they have not just given us loop of Henle, but they have specifically asked, see, proximal convoluted tubule is ruled out, distal convoluted tubule is ruled out pertaining to countercurrent mechanism. But we still have option B and C, descending limb of loop of Henle or ascending limb of loop of Henle. Which one is important for countercurrent multiplier? Here we have commonly told loop of Henle. Let us, as we discuss, you will be able to answer that as well. So the green color structure, what you see is the loop of Henle. Surrounding that, which is the countercurrent multiplier. Surrounding the countercurrent multiplier, this red color one is a vasa recta. Okay, and again look at the direction in vasa recta. Vasa recta is again in opposite direction. Okay, the flow. And then look at the collecting duct, which acts as the equilibrating device. All these three are components of countercurrent mechanism. Now let us see how does this countercurrent mechanism operate. So the green is the loop of Henle, then we have the vasa recta, which is a blue in color, and then collecting duct, which is the equilibrating device, which is in purple. Now you see countercurrent multiplier is the loop of Henle, right? Sorry. Okay, so now how does this countercurrent, how does the loop of Henle multiply? How does it act as a multiplier? So for that we have to understand this. Look at the descending limb of loop of Henle and ascending limb of loop of Henle. The descending limb of loop of Henle is permeable to water. Just remember this one point. Ascending limb is permeable to solute. Okay, if you just remember this, it will be the easiest. That too, which portion? This both you have the thick ascending limb of loop of Henle and thick ascending limb of loop of Henle, right? So this is the thick portion, this is the thin portion. Okay, thin portion it is passive, in thick portion it is active. This also I want you to remember. Okay, with this much of knowledge we will be able to crack the countercurrent mechanism. So we have the loop of Henle. The loop of Henle has a descending limb, then it has a ascending limb. Ascending limb also we have the thin ascending limb of loop of Henle, then thick ascending limb of loop of Henle. What are the two important things which you have to remember? Descending limb is impermeable to solutes, permeable to water. Okay, that means what can happen? Only water can move out of the descending limb of loop of Henle. Similarly, the ascending limb of loop of Henle is permeable to solutes, impermeable to water. So what can move out of this region? Only solutes can move out of the ascending limb of loop of Henle. Even in this ascending limb of loop of Henle, the movement of solutes out of the thin ascending limb of loop of Henle is always a passive process, whereas the movement of solutes across the thick ascending limb of loop of Henle is an active process. Okay, so here it will occur by diffusion. Here it will occur with the help of the sodium potassium ATPase pump. In thick portion, you need a pump to actively transport solutes out of the thick ascending limb of loop of Henle. Okay, now it will be transported out where? This is the medullary interstitium. So all the space what you see in between is the medullary interstitium. So the solutes will be transferred into the medullary interstitium. What is the goal or aim of countercurrent mechanism? To establish a medullary osmotic gradient. Okay, that medullary osmotic gradient is established and the medullary interstitium needs to be hyperosmolar. The medullary interstitium needs to be hyperosmolar. Only if the medullary interstitium is hyperosmolar, we will be able to produce a concentrated kidney. Now, do you understand what is the importance of countercurrent? Why do we need a countercurrent? We saw what is countercurrent mechanism, what are the components. Now, why do you need a countercurrent mechanism or what is the use of this countercurrent mechanism? Countercurrent mechanism helps to establish a uh, medullary gradient and increased osmolarity of the medullary interstitium. Only if the medullary interstitium is highly osmolar, we will be able to produce a concentrated urine. Otherwise, we will produce a dilute urine. That is why our patient is having difficulty in forming a concentrated urine because his countercurrent mechanism is impaired. Okay, so now one of the components of the countercurrent mechanism is the loop of Henle, which acts as a multiplier. Since the descending limb of loop of Henle is permeable to water, the filtrate which comes from the proximal convoluted tubule, it will be isotonic to that of plasma. Now, as the filtrate passes through the descending limb of loop of Henle, it will lose water. And by the time it comes at the tip of the loop of Henle, the fluid is going to be hypertonic to plasma. Whatever is coming initially will be isotonic. As it keeps going down, it will become more and more and more hypertonic. So because the fluid is removed, the removed fluid, where will it come? The water that leaves the thin descending limb of loop of Henle, where will it come? It will come into the medullary interstitium. What is our goal? To establish a medullary interstitium which is hyperosmolar. More solute load should be here. But what is happening? Water is coming and diluting it. Do we need this? No. So immediately what will happen? This vasa recta, okay, this blue color structure, the vasa recta will remove the water from the medullary interstitium. Okay, look, the water is, can you see the arrow? Water is going into the vasa recta. The water which goes into the vasa, this is the ascending limb of vasa recta. So the water which goes into the ascending limb of vasa recta will go into the general circulation. So by this way, the countercurrent mechanism also helps in maintaining the blood volume, fluid volume in the body. So the water which comes out of the descending limb of loop of Henle into the medullary interstitium will be immediately picked up by the vasa recta and that water goes to general circulation. That is one point. Now, the fluid is hypotonic to that of plasma. Now it is going to ascend up first into the thin ascending limb of loop of Henle. As it is ascending up in the thin ascending limb of loop of Henle, compared to the medullary interstitium, this tubular fluid is going to have more solutes. Why? Because it is hypertonic. Automatically, solutes are going to diffuse out from the thin ascending limb of loop of Henle into the medullary interstitium by a passive process. So it is diffusing out. As it is diffusing out, it is losing the solutes. Solutes are getting added into the medullary interstitium. Then it is ascending up into the thick ascending limb of loop of Henle. Once it comes to the thick ascending limb of loop of Henle, still some solutes are remaining, but it is not going to diffuse anymore. Okay, that concentration gradient is lost. Now what is going to happen? The active pump, sodium potassium ATPase pump, is going to again pump the sodium ions and chloride ions. All these, the sodium potassium pump is going to pump sodium, and then other active mechanisms are going to pump the ions from the thick ascending limb of loop of Henle into the medullary interstitium. So collectively, by all this mechanism, what is happening? The medullary interstitium becomes hyperosmolar. How much our ideal goal is to get a medullary interstitial hyperosmolarity of 2200 milliosmoles per liter. At the tip it should be 1200, and it is a gradient, so it will be like slowly 300, 600, 800, like that a gradient will be established. Maximum is 1200 milliosmoles per liter has to be established. So 50% of the contribution comes from the solutes, mainly sodium and chloride. Okay, then the remaining 50% of the contribution comes from the collecting duct, the equilibrating device. The collecting duct will cause the diffusion of urea. Urea transporters will will transport urea from the inner medullary collecting duct. The outer medullary collecting duct is impermeable, so from there nothing can happen. The inner medullary collecting duct, the fluid in the inner medullary collecting duct will the fluid in the inner medullary collecting duct will have lot of urea. Why? Because some urea will be even secreted in the PCT from the peritubular capillary. So overall there is lot of urea. Now this urea will be diffused into the medullary interstitium. So 50% of the contribution comes from sodium chloride ions, 50% of the contribution comes from the urea, which is coming out of the inner medullary portion, inner medullary collecting duct. Okay, whenever urea is coming, it is an osmotically active device. To maintain the osmotic equilibrium, water will also follow. In a similar manner, the water will be taken up into the general circulation by this vasa recta, which is called as exchanger. So by this way, now you know how the loop of Henle acts as multiplier. So by adding the solutes, it is multiplying the interstitial hyperosmolarity. If you closely look here, it is mainly which portion of the ascending limb of loop of Henle which actually acts as multiplier by adding the solute. Is it the ascending or descending? Yes, it is the ascending limb of loop of Henle which actually acts as a multiplier. In descending limb, only the water is moving out, that's all. Okay, so that gives us the answer for our question. So among these four options, two already we ruled out. Which is it? The ascending limb of loop of Henle helps in establishing the osmotic gradient by acting as a multiplier. Okay, that answers our question. Now let's further continue our discussion. So now we know about the multiplier. Vasa recta is the exchanger. Exchanger means what? It is going to exchange the solutes and it is going to remove the water. By doing so, it is maintaining the interstitial osmolarity that has been established by the multiplier. Multiplier has already multiplied and established the interstitial hyperosmolarity, which needs to be maintained. Otherwise, what will happen? If not, it will be washed off because of the high blood flow in the renal capillaries. So, to prevent that washing off, what is happening? The vasa recta acts as the exchanger. What is it going to do? It removes the fluid. Now coming to the ions, what is happening is, if you see the sodium chloride here, the ions enter the vasa recta via the descending limb, then leave the vasa recta via the ascending limb, and this keeps on continuing. Okay, so that recirculation of the solutes keep happening within the segment and nothing is washed off. That is a function of the vasa recta as countercurrent exchanger. And already we saw how the collecting duct acts as the equilibrating device. So once urea is leaving, along with urea, water will also follow. Once water leaves this compartment, the urine becomes concentrated. So that is in gist about the countercurrent mechanism. Okay. Okay. Now we have a 58-year-old patient with chronic obstructive pulmonary disease presenting with worsening of, so today we have seen so many patients with COPD, right? So we have a 58-year-old patient presenting with COPD and worsening of shortness of breath and a history of long-term smoking. Okay. Arterial blood gas analysis shows a pH of 7.34. So he is slightly acidotic. Acidosis is there. And what type of acidosis is this? Respiratory problem, right? See, the partial pressure of carbon dioxide is 55 mmHg. So what type of acidosis is this? Respiratory acidosis. What is the normal level? Anything below 45 mmHg is normal. So here it is again high. And bicarbonate of 28 milliequivalents per liter. So if you look at the normal value, normal bicarbonate concentration is around 22 to 28 milliequivalents per liter. Our patient is having 28 on the higher normal side. Similarly, the pH is reduced. Normal pH is 7.35 to 7.45. In our patient, it's 7.34. So there is acidosis. Partial pressure of carbon dioxide is increased. Normal is 35 to 45 mmHg. He has 55 mmHg. So this patient is having respiratory acidosis. Okay. And to compensate for this respiratory acidosis, the kidneys is trying to save the bicarbonate. So the bicarbonate content is slightly increased. So this is a respiratory acidosis compensated with a metabolic compensation. Okay, based on this finding, which of the following best describes? So it is respiratory acidosis with metabolic compensation. That is the answer. It is a very simple direct question. Okay. Now, if it is metabolic acidosis, what kind of description we can see? Metabolic acidosis. Respiratory means the problem is with the kidney. Respiratory means the problem is with the lungs. Metabolic means the problem is with the kidney, right? So, in case of metabolic acidosis, what is going to happen? There will be acidosis, the pH will be low. The bicarbonate will also be low. When kidney is involved, the bicarbonate will also be low. Partial pressure of carbon dioxide might be normal. So that is metabolic acidosis. In case of respiratory acidosis, just now we saw. Classically, partial pressure of carbon dioxide will be more. Okay. In metabolic alkalosis, the pH will be high. Bicarbonate will be high, partial pressure of carbon dioxide will be normal. In respiratory alkalosis, partial pressure of carbon dioxide will be low. The pH will be definitely high because it is alkalosis. Bicarbonate will be normal or low. Okay, so this is the simplest form of presentation to describe all these four acid-base abnormalities. Okay. Okay. Now we have another patient, 34-year-old patient, presents with polyuria polydipsia. Polyuria polydipsia and normal blood glucose. Okay. Urine analysis reveals glucose in the urine. That means he is having glycosuria. Okay. So he has polyuria, polydipsia, glycosuria, but no polyphagia. Okay, this suggests a problem with reabsorption in the proximal convoluted tubule. Which of the following transport mechanism is most likely impaired in this condition? First tell me, is this patient diabetic or not? Is this a diabetic patient? Yes or no? No, this patient is not a diabetic. Okay, you have certain features of diabetic, but this patient is having normal blood glucose levels. Okay, since his blood glucose levels are normal, that is why he is not having polyphagia. Only thing is he is having is polyuria and polydipsia. Why he is having polyuria and polydipsia? Because of this glycosuria. He is excreting glucose in the urine. Since glucose is an osmotically active particle, water is also lost. So that is why he is drinking a lot of water and he is losing a lot of water. Since he is losing a lot of water, he is drinking a lot of water. Okay, so this is not a diabetic patient, but he is losing glucose in urine. So what is the other reason? Normally will glucose be present in urine? Only in case of diabetes, once it reaches increases more than 180 milligram blood glucose level increases more than 180 milligram per deciliter, it will be glucose will be lost in urine. Otherwise, what is a normal phenomenon? All the glucose that is filtered at the glomerulus will be completely reabsorbed at the PCT. Glucose 100% reabsorption of glucose happens at the proximal convoluted tubule. Now in this patient, because of the problem with reabsorption of glucose in PCT, he is not able to reabsorb the glucose and it is secreted and it is getting excreted in the urine. Okay, which is the transport? Basically, what are they asking? The question is very simple. Which is the transport mechanism that is present in the proximal convoluted tubule that aids in the reabsorption of glucose? That's all, as simple as that. How does glucose reabsorption happen? With the help, so many times we have seen this, with the help of a co-transporter by a secondary active transport mechanism via SGLT. What is this SGLT? Sodium glucose co-transporter. So look at this. This is the renal tubular cell in the proximal convoluted tubule. Okay, on the basolateral side, we have the sodium potassium ATPase pump. So this pump, what it is doing, it will pump sodium ions outside, potassium ions inside, and it creates a gradient for sodium. That is, inside of the cell, the sodium is less compared to the outside. So automatically what will happen? Sodium from the tubular fluid will enter into the cell. Okay, in response to this, sodium from the tubular fluid is entering into the cell. Inside, sodium concentration decreases. To compensate that, sodium from the tubular fluid will enter into the cell. When sodium from the tubular fluid enters into the cell, along with that, glucose is also coming. Okay, this is how glucose from the tubular fluid is completely reabsorbed at the proximal convoluted tubule. And the name of this transporter is called a sodium glucose co-transporter. Okay, once inside the cell, the glucose has to go into the blood. So how is it going to go? By facilitated diffusion with the help of GLUT2, glucose transporter. What I want you to remember here is in the same SGLT is also present in the intestines. Okay, but the one in the kidney is SGLT2. The sodium glucose, sorry, the sodium glucose co-transporter that is responsible for reabsorption of glucose from the renal tubules at the PCT is SGLT2. Okay, that is very important. So here it is SGLT. The answer is sodium glucose co-transporter. The next question. We have a 60-year-old patient presenting with symptoms of bone pain, weakness, increased risk of fracture. So the patient is having osteopenia. All these symptoms are suggestive of osteopenia. He has bone pain, weakness, and increased risk of fracture. Laboratory tests reveal increased serum calcium and elevated parathyroid hormone. Obviously, when parathyroid hormone level increase, what is the action of parathyroid hormone? To increase the serum calcium concentration and decreases serum phosphate levels. It increases serum calcium and decreases serum phosphate. So that is why biochemical tests reveal elevated serum calcium. These findings are indicative of hyperparathyroidism. Which of the following mechanism is primarily responsible for increase in serum calcium in this condition? They are asking in hyperparathyroidism, what is the cause for increase in serum calcium? Okay, so before answering that question, you should know about the functions of parathyroid hormone. So we have parathyroid hormone. It is a polypeptide hormone. It acts at three places. Okay, on the bone, in the kidney, and in the intestine. Okay, what does it do in the basically already I told you, parathyroid hormone is going to increase the serum calcium and decrease the serum phosphate level. How is it going to do? By acting on these three structures. On the bone, what is it going to do? On the bone, it causes bone reabsorption. That means osteoclastic activity. It is going to increase the osteoclastic activity. Basically, it is going to erode the bone. Okay, it's going to cause bone erosion. Okay, then when the bone is getting eroded, what will happen? The calcium level increases. In the kidney, what is it doing? In the distal convoluted tubule, it is going to increase the upregulation of a protein, transport protein called as transient receptor vanillide type protein channel 2. Okay, so now this transient receptor vanillide type protein channel 2 is going to cause reabsorption of calcium. Okay, it's going to cause reabsorption of calcium. So that is also again going to cause increase in serum calcium level. In the intestine, what it is going to do? Indirectly, it is going to activate vitamin D in the kidney. By activating the vitamin D in the kidney, what it is going to do? This activated vitamin D is going to cause absorption of calcium from the intestine. So collectively, by acting on the bone, by acting on the kidneys, by acting on the intestine, parathyroid hormone increases the serum calcium level. At the same time, in kidney, in the distal convoluted tubule, it causes reabsorption of calcium. In the proximal convoluted tubule, it inhibits the reabsorption of phosphate. That is why it decreases serum phosphate, increases serum calcium. So in a patient of hyperparathyroidism, we can always find hypercalcemia. Because of this hypercalcemia, more calcium will be excreted in the urine. So hypercalciuria. Because of this hypercalciuria, they have kidney stones, hypophosphatemia, decreased serum phosphate level, and all this features of osteopenia is going to be there. Why? Because this increased serum calcium happened by eroding the bones, so the bones become very weak and easily breakable. Okay, so now with this background knowledge, now let's approach the options. So in hyperparathyroidism, what is the reason for the increased serum calcium? So many reasons are I told you. Let's see what reason is given in this option. One is it because of the increased calcium absorption? Yes, partly. Is it because of increased renal calcium excretion? Or is it because of bone reabsorption? Or is it because of decreased calcium release from the bone? See, increased calcium absorption from the intestine happens, but that is not direct effect of parathyroid hormone. That is indirectly by activating vitamin D. The direct effect of parathyroid hormone to increase serum calcium level is through bone reabsorption. So the right answer here will be bone reabsorption. Don't be in a haste to select option A. I think they have specifically asked pertaining to elevated parathyroid hormone. Okay. Okay. Next, we have another question. A 45-year-old patient is diagnosed with a condition where there is an excessive production of cyclic AMP, adenosine monophosphate, despite normal level of its precursor ATP. Cyclic AMP is formed from ATP. Now the ATP is in normal concentration, whereas the cyclic AMP is very high. Okay, this abnormal cyclic AMP production is due to a defect in the regulatory mechanism of an enzyme. So the enzyme that degrades this cyclic AMP is deficient or is absent. That is why despite normal concentration of ATP, still the AMP levels are very high. That means, what do you understand? AMP is not getting destroyed, and they are asking which is the enzyme that is responsible for breakdown or hydrolysis of cyclic AMP. So which of the following enzymes is primarily responsible for breakdown of cyclic AMP? And its dysfunction would lead to elevated levels of okay, basically which is the enzyme that is deficient, that is what they are asking. Is it adenyl cyclase, or is it phosphodiesterase, or is it protein kinase A, or it is guanylate cyclase? Okay, so for that let us look at the cyclic AMP and its metabolism. What is cyclic AMP? So this is a cyclic AMP is a second messenger which is synthesized from ATP in the presence of the enzyme adenylate cyclase. So adenylate cyclase is going to form cyclic AMP from ATP. So you have ATP. From this you are going to get cyclic AMP with the help of the enzyme adenylate cyclase. Okay, now this cyclic AMP is degraded by an enzyme called as phosphodiesterase, PDE. Phosphodiesterase is an enzyme that degrades the cyclic AMP. Then what is this protein kinase A? Protein kinase A is the cyclic AMP brings about its second messenger effects by activating protein kinase A. Okay, so what is the answer here? Answer here, the enzyme that degrades cyclic AMP is nothing but phosphodiesterase. Phosphodiesterase is an enzyme that deactivates or degrades cyclic AMP. It is the same phosphodiesterase which degrades cyclic GMP as well. If they ask you for cyclic GMP also, it is the same phosphodiesterase only. But to be very specific, it is phosphodiesterase 5. Phosphodiesterase 5 is the enzyme that degrades cyclic GMP. Okay. And also another interesting fact here is stimulants like caffeine, coffee, the caffeine present in coffee, how does it bring about its stimulatory effect? By inhibiting this phosphod. When we drink coffee, the caffeine in the coffee inhibits this enzyme phosphodiesterase. So when it inhibits phosphodiesterase, more of cyclic AMP is available, which brings about the stimulant effect of coffee. Okay. Okay. Coming to the next question. We have a 62-year-old patient who is a chronic hypertensive and he is being evaluated for an abnormal. So we have an hypertensive with abnormal heart rate response to postural changes. So when the person is changing his posture, the heart rate response is not normal. When moving from what type of posture? When moving from a lying to a standing position, the patient experiences a significant drop in the blood pressure and an inadequate compensatory. So there is a drop in a blood pressure, an inadequate compensatory increase in heart rate. Which of the following mechanism is primarily responsible for regulating heart rate and blood pressure changes with response to postural changes? This is also a very easy question, right? So what is the patient having? He has orthostatic hypotension. That is, when he changes his posture from lying down to standing. When lying down to standing, for all of us, anybody, normally what will happen? There will be venous pooling of blood in the legs because of the effect of gravity. Suddenly from lying down posture, when we become upright, due to the effect of gravity, since veins are the capacitance vessels, there will be venous pooling of blood. Okay, more than 20% of the blood will go into the veins of the lower limb. Because of this venous pooling of blood, what is going to happen? The venous return to the heart decreases. Okay, so the end diastolic volume decreases, the cardiac output decreases, so the blood pressure drops. But we do have compensatory mechanisms that regulates blood pressure. What are the important compensatory mechanisms that regulate blood pressure? We have the short term, intermediate and long term. So here we want the short term mechanisms to immediately act. So under short term, what are the mechanisms? We have three important mechanisms: baroreceptor, chemoreceptor and CNS ischemic response. For correcting the orthostatic hypertension, which is the regulatory mechanism, it is nothing but the baroreceptor reflex. So what is the baroreceptor reflex going to do? It is going to immediately bring back the blood pressure to normal. It is going to cause sympathetic activation, increase the heart rate, increases the cardiac output, increases the blood pressure and brings it back to normal. In this patient, that baroreceptor mechanism is impaired. Okay, so what is the answer here? It is option B, baroreceptor reflex is impaired. Okay. What about this chemoreceptor reflex? Chemoreceptor reflex is going to act via, what is it going to do? It will usually respond to changes in the chemical composition of blood, whether it is hypoxemia or hypercapnia. Then it is going to stimulate the respiratory center, and from there the cardiac center is going to be stimulated to facilitate mainly the gas exchange, right? And also increase the blood pressure. What about renin-angiotensin-aldosterone system? That is for long-term regulation of blood pressure. Again, the sympathetic nervous system will also cause increase in blood pressure. And this baroreceptor is going to act through the sympathetic nervous system. So the answer here is the baroreceptor reflex. And where are these baroreceptors located? Baroreceptors are nothing but pressure receptor located in the arch of aorta and the bifurcation of internal and external carotid arteries. Okay, so at these places only these receptors are located, because these are the regions where there is high blood flow, and they will be able to immediately sense the decrease in blood pressure with the rate of blood flow. Okay, immediately what is it going to do? It is going to activate all those responses I told, and it is going to bring back the blood pressure to normal. Okay. Then coming to our last question, which of the following statement about endocrine functions of the kidney are true? So there we are discussing about the endocrine function of the kidney. So the first option is the kidneys produce erythropoietin, a hormone that stimulates the red blood cell production in bone marrow. Yes, this is true. Renin secreted by the kidneys is crucial for regulating blood pressure through its role in the renin-angiotensin-aldosterone system. Yes, this is also true. The kidneys secrete calcitonin to regulate calcium levels in the blood, working antagonistically to parathyroid hormone. Does it secrete calcitonin? Calcitonin is secreted by what? The parafollicular cells of the thyroid gland. So this is false. Okay. The kidney's endocrine function includes the conversion of inactive vitamin D to its active form, calcitriol, which aids in calcium absorption from the intestine. This is also true. So actually, this question, they should have asked which is false. If they have asked which is false, then it will be option C. Here they have asked to select which is true. All the three are endocrine functions of the kidney. Other than this, other endocrine functions of the kidney are, it helps in acid-base balance as well. Okay, so now if you have to go with one answer, then I would go with erythropoietin. But actually this is a wrong question. It has to be which is false. Okay. So thus with this we are completing our discussion on clinical based questions in physiology. Okay, I wish you very best, all the best, so that you will be able to clear your exams and get your postgraduate subject of your choice. We'll take a break for lunch and we'll come back with image based discussion. Thank you.